Hello, everybody. Welcome to day 3 of 11 days of biochemistry, genetics coming your way. And so we're right where I want to be after 2 days of solid molecular biology. And just of a quick synopsis of what we've done over the last 2 days. We did the first 4 chapters of 7 chapters in molecular biology. Chapter 1 deals with what is a chromosome, what's DNA, that kind of stuff. Chapter 2 dealt with replication, how you replicate DNA, make more DNA. And Chapter 3 dealt with transcription. You got to know that word, transcription. You're going to see a lot coming up in Chapter 5. But Chapter 3 talked about transcription, how RNA polymerase binds to the promoter and turns on a particular gene. And then Chapter 4, which is the bulk of yesterday, dealt with translation, how we take the RNA that came from DNA, how we take the RNA and make a protein from that. And it all depends whether it's a prokaryote or a eukaryotic situation of how that RNA is made and how it is processed, and then how is it translated into making protein. So it's both prokaryote and eukaryote dependent. So we covered all the high yield stuff in the first 4 chapters about basic biochemistry of DNA and RNA. As long as you were paying attention, you don't have to go reading other things. I saw a lot of you were texting each other right now about other resources. As long as you pay attention, as long as you're understanding it, as long as it makes sense. In terms of biochemistry, I can't speak for the other disciplines, but in biochemistry, I got your back. It's all you need to know. Just focus in on the stuff we talk about. When I say high yield, make sure you write it down, maybe a star or something, and that night or as soon as you can, go back over the high yield stuff and master it, not memorize it. And by the way, what I mean by mastering it is you can't stare at something and you can't sit. You cannot be sitting back and reading the book or reading the notes and so on, and just stare at it, and then you think you're going to be able to answer questions on board exams. That's passive. You're not mastering anything if you just simply stare at something and then pretend you're going to give back the information. What I mean by mastering it is you have to verbalize a particular process. For example, if I said to you, why does, like from yesterday's review, the why question, why does eye cell disease present with inflammation and inclusion bodies? Can you explain it? Can you verbalize the answer without looking in a book? And so as you start to explain either to yourself or to a classmate or something, as you start to explain it, as you start to verbalize it, you'll find that you'll start making mistakes, and you'll find that you forgot something, and then go back again, and then keep doing it, reciting, just think about what you're saying, and then after 2 or 3 or 4 times, you've mastered it. Because if you have to verbalize something, then you have mastered the topic. That's teaching. Like all the stuff that I'm talking about in these 11 sessions in biochemistry and so on, I wasn't born with this material. It's simply the reason why I know it is because I have to verbalize it. I have to explain it. And as I try to explain it, I try to do as simple as I can, make it understandable. And I can never make it understandable if I don't understand it first. So try it out. That's why on Facebook, I want you to explain the answer. Just don't put option C or option D. You're not explaining anything. And those of you, even though there's still not a lot of people, I'd like to see more people. Those of you who are putting in the option of a particular question and explaining it, that's great. You're doing exactly what you should be doing, because as you start typing in the answer, you're thinking about it. Oh, maybe I should say this, or maybe I should say that. Oh, I forgot. Oh, that's a mistake. You start thinking about it. Trust me, it works this way. I've had 17 years of experience dealing with medical students and board exams. I know how to do it in biochemistry and genetics. Again, I cannot speak for other disciplines. Other disciplines, they talk about mnemonics and things like that. That's fine. Dr. Barone talks about a lot of mnemonics in pathology, and that's fine. For pathology, maybe it works. But biochemistry, it doesn't work. So on board exams, knowing mnemonics for the Krebs cycle does no good because they're not on board exams. They're not going to ask you what follows fumarate in the Krebs cycle. They're not going to do that. But they're going to ask you, what's the Krebs cycle for? Why is it so important? It is important, but why is it important? Things like that. Explain, verbalize. Just try it. Just try it. Just try it out. You'll see. You'll master the material. Okay. All right. Now, what's coming up next is chapter 5. So in chapters 1 through 4, we did basic molecular biology. And chapters 5, 6, and 7 is modern molecular biology. And I'm telling you right now, they love the modern stuff, the modern molecular biology. Chapter 5 deals with the regulation of eukaryotic gene expression, how you find a gene and turn it on in your chromosomes. Chapter 6 deals with cloning and making millions of copies of a particular gene. And what are you going to do with that gene? So you make millions of copies. So what? What are you going to do with the gene? That's coming up in chapter 6. And then chapter 7 deals with the all-important topic of genetic testing. How can you analyze DNA or RNA or whatever to look at a particular disease trait or looking at forensics in a crime scene? How can you analyze DNA? That's chapter 7. So again, modern molecular biology. Now let me stress here as well. Depending on when you went to medical school and where you went to medical school, and talking to medical students over the last few years, some of the stuff we're going to talk about tonight, this may be the first time you heard about it. But but again, don't make excuses. Don't make excuses. If this is the first time you heard about some of these things, like enhancers and specific transcription factors, those type of things, if that's the first time you've heard about it, is tonight. That's okay. As long as I do my job properly and explain it, what you need to know, then you're going to be fine. Don't panic. Don't worry. I got your back. I'm there for you. But don't panic. Just try to understand because there's really high yield stuff. Now, chapter 5, regulation of eukaryotic gene expression. How do you turn on a particular gene in your chromosomes? Now, in the old books in Kaplan, Kaplan Biochemistry, chapter 5 dealt with regulation of gene expression, and meaning prokaryotes and eukaryotes. And we first started out talking about prokaryotic gene expression. So we spent a long time talking about the lac operon, for example, how you turn on the lac operon in bacteria. But that is low yield nowadays, low yield. So we deleted that from our chapter 5. So the idea is don't worry about the lac operon or gene expression in prokaryotes. It's low yield. I cannot guarantee it that you won't see the lac operon on boards. I've heard about it being on the boards one time in the last 5 years. Take a chance that it won't be on the boards. Focus your attention on the high yield material that is likely to be on board exams, and you'll do fine. So don't worry about prokaryotic gene expression, because after all, you're going to be a doctor of human beings, not a doctor of E. coli, right? You're not going to be a doctor of E. coli. So the idea is you're going to worry about what happens in me and you. All right, so that's what this chapter is all about. How you find a gene in your 46 chromosomes and how does RNA polymerase turn it on. Okay, now what's coming up here is, in this chapter, there are 4 figures that we need to talk about. It's the last 2 figures that are the high yield figures. And I'm telling you right now, extremely high yield. Almost every discipline is in the last 2 figures coming up. The high integration, not just biochemistry with path. It's biochemistry, pathology, pharmacology, physiology, microbiology, all coming up. Immunology, all, coming up in the last 2 figures. The first two figures set the stage for the last two figures. So again, so please pay attention to these preliminary figures, and then you'll start to see, wow, now I see why physiology, pathology, pharmacology are talking about gene expression. You're going to see a major integration coming up. All right, so the first of the two preliminary figures is this one shown on page 74. And now suppose in the red, this represents some gene region, some gene that we want to turn on. So in other words, this what you're looking at here, this is a very small part of a very long chromosome that goes in either direction. And in the red represents some gene region that we want to turn on. And in the green, suppose that represents the promoter to which RNA polymerase will bind and then turn on the gene. Now remember, the gene has plus nucleotides, +1, +2, +3, and so on. The promoter region has negative numbers, -1, -2, -3, and so on. So again, nothing new. So RNA polymerase binds to the promoter and turns on the gene. Now that's what we talked about in chapter 3. Nothing strange here. But if you were to think about it, you would say, man, there's got to be more to the story. Because after all, how does RNA polymerase know which promoter to bind to? You have thousands of promoter. Each gene in your chromosomes has a distinct, unique promoter sequence. How does RNA polymerase know which is the promoter for beta globin, what's the promoter for alpha globin, and so on? How does one enzyme know which promoter to bind to? That's what chapter 5 is all about. How does it happen? Okay, so what's coming up here is the rest of the story to explain how does RNA polymerase know which promoter to bind to. As you would imagine, RNA polymerase needs help, and that's what that's coming up in this chapter. And there are terms called enhancers. And by the way, just to show and illustrate how high yield this stuff is, last year there was a medical student who told me they had 3 questions that dealt with enhancers. So if I were you, I would pay attention to this because you got to know what happens in eukaryotes. Okay, now what happens here is that so here is a gene in the red that we want to turn on, and in the green is the promoter, the binding site for RNA polymerase. Now it turns out within the promoter there are domains, DNA domains, called one of them is called the CAT box, another one's called the UPE-GC rich box, another one's called the TATA box. These are domains within the promoter, low yield. But what these domains serve as, they serve as binding sites for a class of proteins called general transcription factors. Look at the words: transcription, RNA synthesis, factors. So these are transcription factors involved in making RNA, and the word general means they're in basically all cells. So these general transcription factors are in basically all cells. They bind to regions of the promoter along with RNA polymerase, and they are involved in transcription of the gene. Now, because they're in all cells, they're involved in basal transcription. Just not very much transcription going on. They're involved in basal transcription. And on the next preliminary figure, you're going to see some of the names of the general transcription factors. Low yield. Even though you're going to see names, don't worry about the names. That's not important. So general transcription factors are in the nucleus of all cells that have a nucleus, and they bind to the promoter along with RNA polymerase. Much higher yield. That's what the rest of the chapter is going to be about. Are other DNA sequences called enhancers. So by definition, an enhancer is a DNA sequence to which other proteins bind. And these enhancers, again, these are DNA sequences. And some of these enhancers can be far upstream, For example, if here's a gene, here's the promoter right next door, just upstream. Far upstream can be an enhancer region, a piece of DNA. And so enhancers can be far upstream. They can be far downstream from a gene that we want to turn on. But look at the word enhancer. So these are DNA sequences which enhance transcription, not required for transcription, but they're involved in enhancing transcription. In other words, when RNA polymerase binds to the promoter along with general transcription factors and starts to transcribe the gene into RNA, maybe in a nucleus, maybe one copy or two copies of RNA are being made. Not much. It's still being made, but not much. But then when the enhancer regions are turned on, that's coming up. But when the enhancer regions are turned on, it's going to enhance RNA polymerase to transcribe the gene. Instead of one or two copies of the RNA, maybe there's 100 copies of the RNA being made. So therefore, more copies of messenger RNA, more protein being made. You're enhancing transcription. Now in this preliminary figure is another major item. So if the enhancers are DNA sequences, then the idea is, well, what binds to them? And the answer is it's a class of other proteins, and these ones are called specific transcription factors. These specific transcription factors are what you need to know for the national boards. High yield items coming up 2 figures from now. But the idea is, look at the words. Transcription factors involved in making RNA, and they are specific to certain tissues. That's what's going to make them so high yield. The integration with endocrinology coming up 2 figures from now. But these transcription factors are specific to certain tissues. And these are proteins. So these specific transcription factors will bind to the enhancer and enhance RNA polymerase to turn this one gene on. Now another item, just like in the promoter, there are domains, like the DNA domains, like the Tata box, the cat box, which are low yield. In the enhancer regions, there are also domains within the enhancer. And these DNA domains within the enhancer to which the specific transcription factors bind are called response elements. So RE stands for response elements. Responding to what? G for GRE stands for glucocorticoids. C in CRE stands for cyclic AMP response element. E in ERE stands for estrogen response element. Hopefully you're starting to realize, man, this is going to be a huge integration with Dr. Wilson's lectures in physiology. But so all the stuff he talked about and all these hormones coming up, turning on this and doing this in the cell, the biochemical basis is chapter 5. All the stuff that Dr. Wilson talked about regarding estrogen, regarding the glucocorticoids like cortisol and so on, cyclic AMP, remember all this beta receptors and so on, with glucagon. But the idea is that's now the biochemical basis is coming up here. So the idea is when you secrete a hormone, then you're going to get the response in a cell specific to a certain tissue here. So again, okay, so now the issue is, again, this is a preliminary figure. So, so yeah, that's right, Arjun, concentrate. Don't be typing in useless information to your classmates. Think what I'm saying, because this will be confusing if you're not paying attention. That's right, concentrate, concentrate. C, it's coming up, Syed. It's coming up, coming up. It stands for cyclic AMP, but it's coming up a little bit later. I'm just setting the stage. This is a preliminary figure. Don't ask for detail yet. That's what figures 3 and 4 are about. So I'm just setting the stage. All right, so the idea is conceptually, how can these enhancers be so far away from a gene that we want to turn on? Remember, some of these enhancers are far upstream of a gene. Some of these enhancers can be far downstream from a gene. How can they be so far away from a gene that we want to turn on? Okay, now to explain that, now again, pay attention, concentrate. So the idea is, suppose this region right here represents a promoter region for a particular gene. Okay, so this is the promoter for a gene. So RNA polymerase will bind to the promoter and turn on the gene. Nothing new, that was chapter 3. Now far upstream, so here's the RNA polymerase binding to the promoter to turn on the gene. And far upstream can be a enhancer region. And in the enhancer region, what happens is when the specific transcription factor is turned on, how's it being turned on? Something called a hormone. When the specific transcription factor is being turned on by the hormone, that specific transcription factor protein will bind to this enhancer, that DNA will bend, and then RNA polymerase is now enhanced to transcribe the gene to make RNA. It's simply DNA will bend. All right, now here comes the second preliminary figure to illustrate the principle of bending of the DNA. So again, what's happening here is, now suppose in red, or orange, whatever color this is, suppose that represents a gene that we want to turn on. It's sitting in your chromosomes. And in the green is the promoter region for the gene. And in the black outline is the enzyme RNA polymerase. Don't worry about Pol II, that's not important. It is RNA polymerase. That was chapter 3. RNA polymerase binds to the promoter to turn on the gene. Nothing new from what we talked about in chapter 3. What is new is, notice here, there's going to be, as shown by these different shapes, there's going to be a complex of proteins that surround RNA polymerase, that help RNA polymerase to enhance transcription of the gene. All right, now the idea here is, in the promoter, there are these boxes, the TATA box, the UPE-GC rich box, the CAT box, low yield. These serve as binding sites for general transcription factors. And these general transcription factors, notice the names, low yield, NF1, SP1, don't worry about them for boards. But these proteins bind to regions of the promoter along with RNA polymerase, and you get this basal transcription. But then when other proteins, now these are the specific transcription factors, when they're being turned on, for example, in the presence of cortisol, in the presence of estrogen, in the presence of cyclic AMP, C stands for cyclic AMP, then those specific transcription factors then are activated and they will bind to the regions of the enhancer called response element. So now we're getting response to the cyclic AMP. We may get response to cortisol. We may get response to estrogen, for example. And as a result, then by binding of the protein to the enhancer region, that DNA will bend. And notice, a huge number of proteins are now telling RNA polymerase to bind to this promoter to turn on this gene. In other words, now if I could simplify this and try to understand what goes on conceptually, just view RNA polymerase as the queen bee, and these other proteins are the helper bees. The queen needs help. Now you watched Animal Planet or Discovery Channel. You should be watching educational programs like that instead of programs like Sex and the City, things like that. You should watch educational programs. Because there they might talk about a hive, a beehive, and they have the queen bee. But then you have all these helper bees. So the queen needs help. The queen needs help figuring out which promoter to bind to. So the idea here is when the specific transcription factors are activated by hormones, these helper bees are telling the queen bee, hey, queen, come over here. Hey, queen, come over here. Sit down on this promoter, not the promoter over there. Sit down on this promoter and turn on this gene, making little babies, little eggs called RNA. That's what's going on. Now, if you don't like bees, you can view RNA polymerase as the queen ant. The queen ant needs help, and these other proteins are helper ants. And the helper ants are helping the queen ant figure out which promoter to bind to. And once it figures out which promoter to bind, then the gene is turned on. Now notice what happens in the absence of the hormone. When you don't secrete cortisol, when you don't secrete estrogen, for example, in this example, then the specific transcription factor is not turned on. The DNA is not bending. A complex of proteins are not forming with RNA polymerase, and therefore only basal transcription. That's why virtually nothing happens when the hormones are not being secreted. But once one of the hormones is being secreted, it activates the specific transcription factors, DNA bends, RNA polymerase comes in and turns on a particular gene. All right, okay, so you understand what goes on. Now again, if you have questions, use the Q&A pod. Dr. Mahad and Dr. Donner, they're dying to answer questions. They want to help. So if you have questions, please use the Q&A pod because they really want to participate. They're sitting there with their thumbs, oh, I hope students ask me questions because I want to help the students out. So if you have questions, they're delighted. They want to help you out. If they feel there's a question that needs to be directed to me, they'll send it to me, and don't worry about that. So again, okay, now what's coming up next is, so we did the two preliminary figures, and now comes the two of them, two high yield figures, and you must understand. Don't make excuses if you don't understand. It's too hard. I'm going to simplify. You must understand it. All right. Okay, now what's happening here is the first of them is a table. Now look at the table before we discuss the table. Look at the integration here with endocrinology, physiology. Look at the integration with pharmacology. Look at the integration with immunology. Look at the integration with embryology. You're going to see pathology as well. Look at the integration with virtually all the other disciplines here. You must understand transcription factors. Modern molecular biology. All right. Now what we got over here is, on this table right now, are the specific, a sum of the specific transcription factors. Now again, I'm going to when you hear the words specific transcription factor, you say to yourself, it's a protein. It's a protein. A specific transcription factor is a protein in a specific tissue. Focus your attention on that. It's in a specific tissue. It's a protein that's just sitting there doing nothing. But when that specific transcription factor is turned on, it's either in the nucleus or in the cytoplasm, and then if it goes into the nucleus, then it's going to bind to a piece of DNA called the response element, which is part of an enhancer region. So the response element is DNA. It's a DNA sequence, which is part of a larger sequence called the enhancer. Just like the Tata box is part of the promoter. A response element is a DNA sequence, part of an enhancer region. And then after the protein binds to the enhancer, then we get some response. All right. Now the first of the ones we're going to talk about here is extremely high yield, the integration with molecular physiology that Dr. Wilson talked about. And that's these steroid receptors. Now the one I'm going to talk about here is the one for cortisol. And you must understand how cortisol works. from a molecular point of view. Now, you all know the end product from Dr. Wilson's lectures. You know what the ultimate outcome of cortisol secretion is going to do. But what's the intermediate basis? What's the biochemical basis of how cortisol works to do the physiology which Dr. Wilson talked about? So that's what this is about. So the idea is when you secrete cortisol, cortisol will find its receptor, and then the receptor is the cortisol receptor, steroid receptor. The cortisol receptor is a specific transcription factor. Look at the column it's in. One more time. The cortisol receptor is a receptor, yes, but it's also classified as a specific transcription factor. And when cortisol binds to the receptor, it's going to go into the nucleus and look for a piece of DNA, a sequence of DNA called the HRE. And HRE, if I were to ask you what does it stand for, it stands for hormone response element. Hormone response element. Now on board exams, you might see the HRE called by another name, and it's called the GRE. You might see it called the GRE or the HRE. GRE stands for glucocorticoid response element. It's a piece of DNA within the enhancer region. Now what that sequence is, who cares? But it's a sequence of DNA to which the cortisol receptor binds. Okay, and then we get the response to the cortisol and the cortisol receptor. Now on the right side is an absolutely high yield item. The class of protein the cortisol receptor is, it's called a zinc finger class of receptor. Zinc finger class of receptor. So the cortisol receptor it's a specific transcription factor, yes, and it's also classified as a zinc finger receptor. Now let me ask you a question here. Let me ask you a question. Is the Dr. Wilson, did he talk to you about what is a zinc finger receptor mean? What does it mean, zinc finger receptor? Did Dr. Wilson discuss what a zinc finger receptor is? Okay, but you must know what it means. And on the next figure, I'll show you why it's so important to know. But in any case, suppose this fist represents the cortisol receptor that's sitting in your cytoplasm of your hepatocytes right now, doing nothing. Just picture, look at your abdomen right now. Just picture in your hepatocytes of your liver, in the cytoplasm, there's the cortisol receptor doing nothing. But when you secrete cortisol, cortisol is going to come into the cytoplasm of your hepatocytes. And when the cortisol sees its receptor, the cortisol receptor, cortisol will bind to the receptor. The receptor undergoes a conformational change and opens up in fingers. And in the fingers, there's zinc. That's why they call it zinc finger receptor. When there's no cortisol, it goes like this. When there's cortisol, undergoes a conformational change and opens up in the fingers. So that's why it's called a zinc finger receptor. And then what happens? It's not cortisol and it's not the receptor individually. Together, when cortisol docks with its receptor, together, they then go marching into your nucleus of your hepatocytes. They go into the nucleus, and get this, they go into the nucleus and they finger all 46 chromosomes. Each and every chromosome is fingered. Remember all that euchromatin where the DNA is exposed? Remember that on page 12? We talked about that on Monday. When in euchromatin, when DNA is exposed, then the cortisol receptor and cortisol together, they finger all 46 chromosomes, specifically in the euchromatin portions, looking for a piece of DNA. And when it finds that piece of DNA called the HRE, then the cortisol receptor then sit down on the DNA bends, RNA polymerase comes in, and you turn on a particular gene. It's amazing how that happens. So it is truly amazing. Every single chromosome is fingered by the cortisol receptor. The X chromosome, the Y chromosome if you're a male, chromosomes 1 to 23. They're all fingered in the euchromatin portions because DNA is exposed looking for a piece of DNA. And once it finds that piece of DNA, don't worry about what chromosome number it's located, but once that piece of DNA is located, it docks. All right, now, and then a particular gene is turned on. Now, this is so high yield, molecular physiology, related to the biochemistry. We're going to talk about it again on the next figure. So the next figure is more information about what I just discussed regarding the cortisol and the cortisol receptor. If you want to understand all that endocrinology, stay tuned to the next figure. All right, now what also happens, yet another specific transcription factor is it's called the Krebs protein, which stands for cyclic AMP response element binding protein. This is yet another specific transcription factor. And when a particular hormone is secreted, like glucagon, in endocrinology, when glucagon is secreted, one of the things glucagon can do is turn on a specific transcription factor called Krebs. Once the Krebs protein is turned on, it goes into the nucleus of the liver cells looking for another piece of DNA called the CRE. Now see it, there we go, CRE, and which stands for cyclic AMP response element. And then we get the response to the cyclic AMP, which in turn responds to the hormone. And then you get some gene being turned on. Now the protein class that the Krebs protein is, it's called a leucine zipper, low yield. Leucine zipper, don't worry about it, but it's where two regions of the protein zipper up and becomes activated, but that's low yield. So leucine zipper motifs, low yield, zinc finger, extremely high yield. As I'll point again on the next figure coming up in a little bit. All right, now these two here, cortisol and its cortisol receptor, as well as the Krebs protein, we'll talk more about them, making sense of this information on the next figure. Now continuing on, now you're going to hear about this later on in pharmacology as well as pathology. There are a class of other specific transcription factors called the PPARs, which stands for peroxisome proliferator activated receptors. Peroxisome proliferator activated receptors, PPARs. Now there are a variety of these PPARs, but only two of them you have to know for the boards. One of them is called PPAR alpha, and the other is called PPAR gamma. Now the beta and so on, not important for boards. But it's these two that are important because of the integration with pharmacology. Now what happens is when the PPARs are being stimulated, then it goes into the nucleus looking for a piece of DNA called the PPREs, which stands for peroxisome proliferator response element. Peroxisome proliferator response element. Now, why is it so important? Because these PPAR transcription factors can be turned on by certain drugs like fibrates, clofibrate, and the thiazolidinediones. Now, here it comes. Now, several weeks from now, you're going to have Dr. Harris talking about pharmacology. You're going to love Dr. Harris coming up later. But let me see if you can recall. And if you have the answer in your book, don't type in the answer. Just try and see if you can recall. That's why sometimes I don't want you to use the public chat pod to put in the answers unless I ask you, because you might be giving away an answer that I'm going to ask. But in any case, let me see your answers here. In pharmacology and pathology, for what reasons, global reasons, do you administer either fibrates or thiazolidinediones? For what reason? What reason? Diabetes, that's true. That's true. But even a more global reason. Yes, you do give it for type 2 diabetes, that type of stuff. But a more global reason why you give these drugs is because to lower serum triglycerides. That is why. Not so much the diabetes. It's simply that obesity can cause diabetes. And so we'll talk about that later on in metabolism. But the idea is that an obese person will have a tendency to have diabetes, and so you give them either fibrates or thiazolidinediones to lower serum triglycerides, and hopefully they will overcome the diabetic effect here. All right, now this is what you learn in pharmacology. Is you give fibrates and the thiazolidinediones to lower serum triglycerides. But that's what they tell you in pharm. But what's the reason for this? What's the biochemical reason how these drugs work? And here it comes, the answer. Because fibrates, when you give a fibrate, what it does, it activates PPAR alpha. When you give thiazolidinedione, it activates PPAR gamma. And by activating either of these two specific transcription factors, notice the biochemistry. These specific transcription factors are now turned on. They go into the nucleus and look for a piece of DNA called PPREs. And importantly, certain genes involved in lipid metabolism are now being turned on. What genes? What's this got to do with lipid metabolism? Look at the words. Peroxisome proliferators. Okay, here comes the bigger picture. Now remember from histology, integration with histology. Remember, what's a peroxisome to begin with? A peroxisome is a very tiny organelle. And a peroxisome is involved in a few processes. One of the processes a peroxisome is involved in is oxidation of very long chain fatty acids here. And so the idea is by turning on certain genes, you're going to proliferate the peroxisomes. You're going to make more peroxisomes in a cell. In other words, in one cell, I'll just throw out a number. Suppose in one cell we have 5 peroxisomes. But now we proliferate the peroxisomes. Instead of having 5 peroxisomes in a cell, we may have 100 peroxisomes in a cell. Because there's more peroxisomes around. Peroxisomes, remember, because they oxidize fatty acids, they are like little furnaces that burn carbon, burn fatty acids. So now if you're proliferating the peroxisomes, proliferating the furnaces need fuel to burn. Where is the fuel? The fuel is out there in the bloodstream, serum triglycerides. So sure enough, the serum triglycerides are brought out of the bloodstream, come into the cell, into those little furnaces called the peroxisomes, and they get burned in the peroxisomes, helping to lower serum triglycerides. That is why. Don't worry about the gory details. Just understand. Make sense out of it. It should make sense. One more time. By giving the drug, you're turning on PPREs, which then go into the nucleus telling RNA polymerase turn on genes involved in peroxisome proliferation. So by proliferating the peroxisomes, more furnaces, and then they need fuel to burn, and serum triglycerides are the fuel. They come out of the blood into the peroxisomes to be burned. Yeah, that's right. That's right. I'm on. Yeah, look how beautiful that is. That's amazing, right? Now, by the way, it's a little bit more complicated with PPAR gamma. PPAR gamma also does something else. What it can do is when it's being turned on, it enhances transcription of genes involved in differentiation of pre-adipocytes to adipocytes. So just pick now. If you say to yourself, well, why would that help you lower serum triglycerides? Now, just here it is. If you're differentiating pre-adipocytes by turning on genes by PPAR gamma with thiazolidinediones, by differentiating pre-adipocytes to adipocytes, again, what's an adipocyte? It wants to store triglycerides. So a lot of room in the adipocyte, and where are the triglycerides? They're out in the blood. So they come out of the blood into the adipocyte for storage, lowering serum triglycerides. Beautiful how this works. Beautiful. All right, now let me say something else, because sometimes students ask about this. The main organelle for burning lipid are the mitochondria. That's the main. We'll talk about that later on. Fatty acid oxidation. That's mitochondria. Now you might ask, well, why don't you give a drug that proliferates mitochondria? But you can't. There is no drug that does that. Remember, mitochondria, they're very complex organelles. Two membranes in them, and they have DNA. Remember, there's mitochondrial DNA. You cannot proliferate mitochondria. You think you might, but you can't. And remember, mitochondria come from your mother. Remember mitochondrial inheritance? But the idea is you cannot proliferate mitochondria. But what you can do, pharmacologically as well as biochemically, is to proliferate the peroxisomes. That is why. So when Dr. Harris is here in a few weeks, and he starts talking about thiazolidinediones and the fibrates, you know exactly what he's talking about because you have mastered it before he comes. So when he's talking about it, I'll talk about it from a pharmacology point of view, how much to administer, how long does it last, the fibrates and so on, all that kind of stuff. I mean, that's what pharmacologists do. You know, I mean, after all, what's a pharmacologist? A pharmacologist is a biochemistry wannabe. I mean, they want to be a biochemist, but the pharmacologists, they're always whining. You ever talk to a pharmacologist? They're always whining about things, whining. And us biochemists, we say, okay, you could talk about this, but because they know the biochemical basis of their profession, pharmacology, is biochemistry. They'll never admit to it, but they know that biochemistry is the basis of it. So again, when Dr. Harris is here, he'll talk about how much to administer and so on. We have to give them something to do, and all that pharmacokinetics and all that. But you know the reason why you give the drug. Now moving on, in immunology, when Dr. Muscatello was here, yeah, you may have talked about it in physiology as well, but in immunology, Dr. Muscatello likely talked about NF-kappa B. I'm not likely, she did talk about NF-kappa B. Now, as you all know, when NF-kappa B is turned on, what it's going to do is bind to DNA sequences called KB elements, and importantly, genes are being turned on involved in inflammation. Now, it says here that the domains are called REL domains, low yield. What's high yield is NF-kappa B, is a specific transcription factor. It's a class of specific transcription factor proteins. It binds a KB element, and as a result, you turn on genes involved in inflammation. Now, let me ask you this: in immunology as well as in pharmacology, what's the class of drugs that's often used to treat inflammation? What's the class of drugs used to treat inflammation? Yes, NSAIDs and all. What's another class of drugs? What another? That's right, the glucocorticoids. So the question is why? Why do you give glucocorticoids to treat inflammation? And the reason is the biochemistry is that the glucocorticoids, what they do is they inhibit the signaling. They prevent the NF-kappa B from binding to the KB elements and preventing the signaling that goes on to turn on the genes involved in inflammation. So when the glucocorticoids are present, those genes are not expressed into RNA and therefore into protein. There it is. Don't worry about the detail. Just think concepts. Make sense of it. Now at the bottom of the figure, I'm going to go in a little bit more detail here. And that's these things called the homeodomain proteins. Now, this has a lot to do with molecular embryology as well as pathology coming up here. Now, what's happening here is in molecular embryology is there are a series of specific transcription factors called homeodomain proteins. Now, what's this all about in terms of a concept, as simple as I can explain? What's this all about? And the idea is, you probably never thought about this, but when you and I were being developed as a fetus, how did it happen that these 5 fingers appear on my right hand, my right arm, of my right shoulder? How did these 5 fingers know where to go? How did these 5 fingers know where to go on my left hand? How did my toes know where to go when I was being developed as a fetus? How did my appendages know to go? Where did my ears know to go between on the sides of my head? How did it happen? You know it happens, but how did it happen? All right, that's the topic here, because what happens there is there is a series of genes that are going to be turned on at the right time of fetal development. Now, what these genes are not important, but there are a series of genes that have different promoter sequences, and these genes can be on different chromosomes. Don't worry about them. But these genes, without naming them, they're on different chromosomes, and they have different promoters. And these genes are being turned on at the right time of development. In a short window in time in fetal development, they need to be turned on because these genes encode for enzymes that place where the fingers and the toes are supposed to go. And so the idea is, well, what turns them on at the right time of development? Now, to explain this another way, use analogy. In your room right now, look in your room right now, wherever you're at, look in your room. Look at all the things powered by electricity. Look at all the lights in your room, the computer in front of you there. You may have a telephone, and you may have a printer in your room, a clock, lamps, and so on. I can come into your room right now and turn on a switch, and all of those things come on at the same time. So that's the idea right now. What is the master switch that turns on all of these genes at the right time of fetal development? Okay, now to explain this, what happens is these different genes with different promoters. What happens is they have the same enhancer sequence. Whatever that DNA sequence is for the enhancer, it's the same. So it might be on different chromosomes, but the idea is the same DNA sequence, and that's for these genes. All right, so now what's the master switch? There are several of these, and one of them is called the homeobox gene, sometimes called the HOX gene. There's a related gene called the PAX gene, other ones as well. Don't worry about the gory details. Understand the concept. There is these are the master switches, and when they get turned on at the right time of fetal development, obviously they're being turned on hormonally. So if the HOX gene gets turned on or the PAX gene gets turned on, it's going to make an RNA for a protein classified as the homeodomain protein. Look what column it's in. Homeodomain protein is a specific transcription factor. So suppose now 100 homeodomain proteins are being turned on from the RNA, from the gene. One of these homeodomain proteins will bind to this enhancer, turning that gene on. Another homeodomain protein binds to that enhancer, turning that gene on, enhancing the transcription of the gene. This another homeodomain protein binds to this enhancer, turning that gene on. So just envision. So multiple homeodomain proteins bind to these multiple same enhancers and turning on all of these genes at the right time of fetal development. It just makes sense what goes on here. So they're all being turned on. And in fact, these master switch genes, as well as these other genes that do the placement of the fingers and the toes, these genes are turned on only one time in your life. Only one time in your life are these genes in euchromatin regions of the chromosomes. You still have these genes in your chromosomes, but they're in heterochromatin, never to be turned on for the rest of your life. The only time they're being turned on is some short window in time in a fetal development situation. Just understanding what goes on here. All right, now what happens here is you got to know this stuff, this molecular embryology, because this also integrates with pathology. This was on the national board. Because now just picture is any gene can be mutated. Yesterday we talked about DNA mutation. Any gene can be mutated. There's 20,000 genes thereabout, and there's 7 billion people in this world, you can imagine any gene can be mutated. And sure enough, there are people in this world who have a mutation in either the Hox gene or the PAX gene. Now here it comes. See, Andrew, this is why I didn't want you to mention, type in the response, because I'm going to ask, what is the name of the disease? Now, without looking at Andrew's answers and so on, what is the name of the disease? I'm trying to get you to recall. What is the name of the disease and pathology in which the PAX gene can be genetically defective? Again, this was on a national board. You got to know it. So what's the name of the disease in which the PAX gene is mutated? It's okay, Andrew. No, it's okay. No, don't. No. I know you want to share information. That's great because you're trying to help, and that's great. But just wait till after I ask the question, then go ahead and respond. But I do want you to interact. Please, I want you to interact. But there's reasons why I do what I do. What's the name of that disease? Anybody know? Okay, and the answer is, it's called, last chance. What's the name of the disease? That's right, Simonfanthi, that's right, and it's called Klein-Wardenberg syndrome. Now, without doing a Google search right now, let me ask you a question here. If you see a vignette on board exams that deals with Klein-Wardenberg syndrome, what do you expect to see? What makes sense to you? If a child is born with Klein-Wardenberg syndrome where the PAX gene is defective, what do you expect to see in the vignette that gives you the clues that it's Klein-Wardenberg? What do you expect? That's right. Hey, well done. See, well done. The best example, the best clinical manifestation is limb abnormalities, right? And if you go to page 79 in the margin of your book in biochemistry there, you will see a little blurb about Klein-Wardenberg. Among all the clinical manifestations, somewhere you'll see limb abnormalities. And there it is. Here comes. It makes sense. And that was on the boards. You know, the student said they got Klein-Wardenberg. He says he got it right because he saw the phrase limb abnormalities here. All right, now another item here. Another item. One more item, then we'll take the break. I know I'm going a little bit over here before the break. But the integration with pharmacology. Huge integration with pharmacology. Many years ago, in the late 1950s, early 1960s, there was a drug that was used to treat pregnant women. It was used to treat morning sickness in pregnant women. And so when they gave that drug, what happened was that the infants that were born to those pregnant women who were given that drug, those babies were born with limb abnormalities. Here it comes, and everybody's ready. Look at that. I'm looking over to the side. Look at that. Everybody. Here comes the question. What was the name of the drug? That's right. Everybody's saying thalidomide. Now, at the time, they quickly banned the use of the drug. Now, at the time, we didn't know why that thalidomide caused the limb abnormalities. We know that it did, but we didn't know the biochemistry. But now, because of modern molecular biology, we know the biochemical basis of thalidomide. And what thalidomide does is it prevents the homeodomain proteins from binding to the enhancer. So just put a big X somewhere along here. So the idea is so in the presence of thalidomide, the homeodomain protein cannot bind to the enhancers. Therefore, those genes are not being turned on, limb abnormalities. In one case, it's pharmacology with thalidomide. In the other case, of pathology, that's Klein-Wardenberg syndrome, the limb abnormalities. There it is. It just makes sense. Okay, now what we're going to do is we're going to take a 10-minute break and then come back. And when we come back, we're going to get to another high-yield figure, the major integration at the top of this table here with cortisol receptor and the Krebs protein. So let's take a 10-minute break, and then I will be back in 10. Welcome back, everybody, for hour two. And let me just okay, we're ready to go. So what we've done now is talk about these specific transcription factors. Now, again, I'm hoping you're looking at this and you're saying, now I can understand what Dr. Turco is talking about. That he said this stuff is high yield. I can understand why it's high yield. Look at the integration, how these specific transcription factors, which are proteins, and they bind to DNA, and specifically to pieces of DNA called the response element, which is part of a larger sequence called the enhancer sequence. So again, just know, try to understand why this is high yield. And again, if you're motivated to be positive, stay positive. What's this got to do with helping me be to pass the board and to be a better doc? Is because you're understanding why you do things, why you give a certain drug like thiazolidines, why cortisol does what it does, and what goes on in what thalidomide does. You understand the pharmacology and the pathology if you've got some basic understanding of the biochemistry. So again, I say it over and over, but I'm just trying to, I know a lot of students have mental blocks. They want to know details. They want to know all that gory stuff, and they lose sight of the bigger picture. Me, I like the big picture first. Tell me what I need to know and help me understand why it's important to know. All right, now if you think page 76, this table is high yield, and it is high yield, wait to the next figure, because the next figure is now a huge integration, a much more high yield figure dealing with integration of biochemistry with physiology, and this integration with this thing called life. Because on the next figure, we're going to talk about a part of what life is all about. I told you I'm big picture, right? I told you I'm a big picture type of guy. And what we're going to talk about right now is part of life. Now, what I mean by this is, again, make it simple. Make it simple. In your body right now, you have all these cells. And in your cells, you have all these metabolic pathways. Pathways to make glucose, pathways to burn glucose, pathways to make fatty acids, pathways to burn fatty acids, pathways to make cholesterol, pathways to make heme. You got all these different pathways in every cell. You may not like to think about it, but you and I are a bag of chemicals. We are a bag of chemicals. And these pathways, these metabolic pathways, we'll be talking about next week, these metabolic pathways then convert this molecule to that molecule, to this molecule, and this chemical. It's all these metabolic pathways. And it makes no sense in this big picture of life. It makes no sense that all these metabolic pathways are working at the same time. It would make no sense in one cell to make glucose and burn it in the same cell. It would be futile. It makes no sense in one cell to make a fatty acid and in the same cell burn the fatty acid. It would be, remember, biosynthesis costs energy. Burning fatty acids, you gain energy. It'd be futile. So the idea is, what controls these pathways? And the answer is as simple as I can state. It's this discipline called endocrinology, part of physiology. Endocrinology. In other words, the hormones. Did you ever ask, why do you have hormones in the first place? A simple question like that. Simple. I'm a simple guy. I ask simple questions. Why do you have hormones? Why do I have to worry about a discipline called endocrinology? And the answer is because hormones are, I like to view the hormones as the conductors of the metabolic orchestra of the body. It's the hormones, when they're being secreted, that come to a cell of a tissue and tells that cell, turn on this pathway, the pathway over there, turn it on. Turn on this pathway over here, that pathway over there, turn it off. It's the hormones are the conductors of the metabolic orchestra of the body. That's what hormones do, as simple as I can state. And an example of this is right here. This is an example of what I just said. How the hormones are the conductors of the metabolic orchestra. The hormones direct the show of the body. The hormones direct the show of the human body. They're telling which pathways to turn on and turn off, and an example is right here. In case you never ask a simple question, when Dr. Wilson's here talking about all these hormones, why do we have them in the first place? And the answer is because they direct which pathways are on and off. As simple as I can state. All right, now the example we're going to talk about here, all those hormones, all those hormones you talk in endocrinology, they're directing metabolic pathways in some fashion. And the biochemistry of how those hormones work, some of them are going to be shown right here. Now, and you'll see how high yield this is. Now, the idea is, suppose this represents, in this example, suppose this represents a liver cell, hepatocyte. And under conditions of hypoglycemia, when the blood glucose drops, you respond to that, and you start to secrete hormones like cortisol and glucagon. Now, before we talk more about that, I got another simple question. I'm a simple guy. I'm going to ask you another simple question. By the way, if you know the answer, don't answer, because I'm going to give you the answer later. Stay tuned. Don't worry about the answer here. But I'm a rhetorical question. I'll give you the answer later. But in your entire medical career, did you ever ask someone, why does a body care how much glucose is in the bloodstream? And I think about that. Glucose is one of the very few substances in the bloodstream. The body cares how much is there. And when the blood glucose is too high or too low, the body sends out hormones to fix the problem. When the blood glucose is too high, it sends out insulin. When the blood glucose is too low, it sends out cortisol, glucagon, epinephrine if you're running, ACTH. And so hormones are sent out from the body to fix that problem. What about other molecules in the bloodstream? The body doesn't care. Glucose is one of the very few substances in the bloodstream. The body cares how much is there. Does your body care how much amino acids are in your bloodstream right now? Nope. Does your body care how many fatty acids are in your bloodstream right now? Nope, it doesn't care. Does your body care how much cholesterol is in your bloodstream right now? Nope, it doesn't care. Does your body care about other sugars in the bloodstream right now? Nope, it doesn't care how much galactose is in the blood. It doesn't care. But glucose, it absolutely cares. And it sends out all these hormones that fix the problem. Now, then later on, not now, but later, I'm going to ask you why. In your medical career, you got to ask why of certain things. And here's that. Why does the body care how much glucose in the blood? And another issue here is, related to this is that when the blood, now if you think in the brain, no, it's not the brain. Good try, but it's not the brain. The brain can use ketone bodies. Now don't answer. That's coming up later. So I'm not going to look at the public chat pod because I don't want to look at the answer. I'm just trying to get you to think. I want you to think. If I just gave you the answer, I want you to think about it. Because you'll start to think, oh yeah, why? Why does the body care? And you start thinking about it, and maybe you come up with the answer on your own without me giving you the answer or somebody else giving you the answer. Because it's part of the learning process. Thinking. I told you I'm going to try to get you to think and not memorize. So just think. We'll give the answers next week. Why does the body care how much glucose is in the blood? Now, another item here related to that. So when the blood glucose drops, the body sends out glucagon, cortisol, ACTH, epinephrine, norepinephrine. Did you ever ask in endocrinology the why question? Why do you have different hormones to do the same thing? Glucagon raises blood glucose. Epinephrine raises blood glucose. Cortisol raises blood glucose. Why do you have different hormones to do the same thing? Stay tuned. It's all coming up later. But the idea, again, start thinking, oh yeah, why? Dr. Wilson talked about all these hormones raising blood glucose, and why? Yeah, that's true. It's part of the thinking process here. But the answer will come later. Again, big picture. Always think big picture. All right, now the idea is that when blood glucose drops, the body says, you know, we don't want to die, so we better fix that problem. So the body sends out hormones like cortisol and glucagon. And what cortisol is going to do, because cortisol is a lipid hormone, a steroid hormone, because it's a lipid, it can go right through the plasma membrane, permeable to the plasma membrane, and comes into the cytoplasm of the liver cell. And when it comes into the cytoplasm of the liver cell, it sees its receptor, shown here in the V. So the V represents the receptor for cortisol, the cortisol receptor. Now let me ask you this, because sometimes students never thought of this, is that when cortisol comes into the cytoplasm of the liver, does cortisol also go into the cytoplasm of the muscle cell? Yes or no? Let me see your answer here. Does cortisol also go into the cytoplasm of the muscle cell? Absolutely, yes. Yes. Cortisol goes into every cell. It goes in and out, in and out, in and out of cell because it's permeable to the plasma membrane. When it comes into the muscle cell, it comes into the cytoplasm. It says, hey, cortisol receptor, are you home? Anybody there? Cortisol receptor? Not here? Okay, I'm going to go back out. And so cortisol leaves the muscle cell, goes to another muscle cell and says, hey, anybody there? Anybody, any cortisol receptor? No? Okay, see ya, goodbye. And eventually finds it in the liver cell. So it goes in and out of cells looking for the cortisol receptor, and it's only found in the cytoplasm of the hepatocyte. All right, now here comes a major item. Man, everything's major, right? Here comes another major item. And again, now, without looking on the previous page, the cortisol receptor, what class of receptor is it again? What class of receptor is it without looking? What class of receptor? It's a steroid receptor, but what class is it? That's right, it is a zinc finger receptor class. Okay, now before we move on, you should all get this right because you had Dr. Wilson in physiology. Here comes the question for you. All right, here comes, get ready. In physiology, or endocrinology, I should say, part of physiology, what's another example of a steroid hormone besides cortisol that uses a zinc finger class of receptor besides cortisol? What's another example? Besides cortisol, what's another steroid hormone that uses zinc finger class of receptor? And you, thyroid hormones, they're not steroid hormones. They're not steroid hormones. I'm looking for another steroid hormone that uses a zinc finger class receptor. I mean, thyroid hormones are not steroid hormones. And the answer is simple. The answer is simple. Another example of a steroid hormone that uses a zinc finger class receptor, the answer is all steroid hormones. Whether it's cortisol, whether it's estrogen, whether it's testosterone, progesterone, all those ones, even aldosterone. You know, aldosterone has to do with the salt balance, but it's working at the gene level to affect the salt balance and so on. So the idea is all steroid hormones use a zinc finger class of receptor, which is also a specific, look at the word, specific to a certain tissue. So the zinc finger receptor for estrogen is a specific transcription factor, specific to a certain tissue. The testosterone receptor is a specific transcription factor, specific to certain tissues. The cortisol receptor is a specific transcription factor, specific to a certain tissue. Again, concepts here. Starts to make sense. All right, now, coming up next. Because you know other things are coming. How do you know that? Because there's more space here. What's another example of a lipid hormone that uses a zinc finger class of receptor? Another example of a lipid hormone. Now, Ayman and Anju, now I need you. Another example of a lipid hormone that uses a zinc finger class of receptor. Ah, we go. Say, yeah, that's right. That is the thyroid hormones, T3 and T4. They use zinc finger class receptor. Now here it comes. Besides the steroid hormones, besides the thyroid hormones, what's another example of lipid hormones that uses a zinc finger class receptor? Another example. And the answer is the vitamin A and vitamin D. Now, Funmi, you said vitamin A, D, E, and K. No. Two of those four. Two of the four are hormones, vitamin A and vitamin D. Vitamin E and vitamin K are not hormones. So they're not lipid hormones. So please, don't memorize. You're trying to memorize, and memorizing doesn't make sense. If you try to memorize, you're not making sense. It's vitamin A and vitamin D are lipid hormones. They turn on gene expression. They're lipid hormones. All right, now on this box right here on the left side of the figure, you can see why I'm emphasizing this. What I'm driving at is, remember in endocrinology, you have two classes of hormones. You have the lipid soluble hormones, the fat soluble hormones, and the water soluble hormones. Now, in terms of the fat soluble hormones, lipid soluble hormones, all of them, every single lipid soluble hormone uses a zinc finger cluster receptor, but it's specific to a certain tissue where the receptor is located. And so they're all going to work basically the same. But what genes are being turned on and so on, where it works, it depends on what hormone that we're talking about. But the initial biochemistry is all the same. Please understand this. All lipid hormones use a zinc finger class of receptor, which are also specific transcription factors. High yield. That's why the National Board expects you to know this here. All right, now coming back to the cortisol receptor. So when cortisol comes into the cytoplasm of the liver cell, it sees its receptor and binds to the receptor and then undergoes a conformational change, opening up in fingers. And then cortisol and cortisol receptor together then go marching into the nucleus of the hepatocyte and then finger all 46 chromosomes, looking for a piece of DNA called the enhancer region, which is specifically a region of it. Part of it is called this GRE, which stands for glucocorticoid response element. Again, you might see it called the HRE, hormone response element. What that DNA sequence is, who cares? But it's a name of a DNA sequence to which the cortisol receptor binds. Now we'll talk more about that in just a minute. Now, when glucagon is secreted in response to low blood glucose, then because glucagon is a polypeptide, it cannot enter the cell to turn on gene expression. So what glucagon does is its receptor is located on the plasma membrane, shown here in the Y. So that is the glucagon receptor. We'll talk more about it later on. But the idea is downstream, it activates cyclic AMP. Now, you know there's a lot of things missing here that we'll talk about later on in biochemistry. G proteins, on adenocyclase. Don't worry about it now, just the concept. I'm trying to get you to understand. But when cortisol binds to the receptor, downstream activates cyclic AMP, which in turn activates protein kinase A, major enzyme we'll talk about later. Don't worry about it now. And then that activates the CREB protein, which stands for cyclic AMP response element binding protein. And once that's specific, it's a specific transcription factor specific to the hepatocyte. And then when that protein is turned on, it binds to that same enhancer region, but another part of the enhancer called the CRE, cyclic AMP response element. And then what's going to happen? So when these proteins dock with the enhancer, the DNA bends, RNA polymerase comes in and binds to the promoter for a major gene in the pathway of gluconeogenesis, that genesis of glucose. Again, notice in response to low blood glucose, now we're turning on a gene involved in making glucose in the hepatocytes. So the gene is now turned on, it's enhanced, making many messenger RNAs. So the messenger RNA is made. Ultimately, the messenger RNA then leaves the nucleus, goes out into the cytoplasm. That's where the ribosomes are located. Then we turn on, we translate the RNA into the enzyme PEP carboxykinase. Don't worry about the enzyme now. That'll be discussed later on. When PEP carboxykinase is turned on, it takes a substrate, makes more product. And by making more PEP, we make more glucose. And by making more glucose in the hepatocyte, it exits the hepatocyte, gets out into the bloodstream to try to maintain blood glucose. There it is. Man, that works. Look how beautiful this is. Beautiful this is. How the hormone is directing the show in the liver. The beautiful how it's turn on gene expression here. Turn on gene. Example of hormone stimulated transcription. Now, if this was testosterone or progesterone or so, that's just different tissues, but they're turning on. This is the same mechanism, but another gene is being turned on by RNA polymerase. But again, it's the concept. Make sense of it. Understanding this. All right, now part of the big picture as well. Now, on the right side over here, I'm going to show you a term. And I know a lot of you, when you see this term, a lot of you are going to cringe. You're going to go, ooh, man, that one hurt. Here it comes. Trust me, I know this is going to happen with some of you. Here it comes. What's happening here is part of the big picture of life with these hormones. Your body is manipulating a term called, here it comes, and the term is called Vmax. You got that? That's right. Vmax kinetics. Ooh, oh. Vmax kinetics. Now let me say something about kinetics. You must understand kinetics. I know medical students hate kinetics generally because they don't understand it. It's not taught properly in medical school. People worry about all these equations and all these Lineweaver-Burke plots and all these Michaelis-Menten. Forget about all that stuff. That stuff is not that important. It's the concepts. You must understand the concepts. Now later on, once we get to kinetics in biochemistry, I'll talk about it only from a conceptual point of view. There's going to be no mathematics. There's not going to be any equations. It's understanding concepts as illustrated right here. Notice, now Vmax, what that means is velocity means product per time. Don't get bogged down. Don't worry about details. Velocity means product being made per time. By having enzymes around, by increasing the number of enzymes, PEP carboxykinase, by increasing the number of enzymes, what are you doing regarding velocity here? Are you increasing Vmax, decreasing Vmax, or no change? Let me see your answer. By having more enzymes, what are you doing regarding glucose being made per unit time? Increase, decrease, or no change? That's right. That's right. It's simple. It's simple. It is increasing velocity, increasing the amount of glucose being made. Now let me stress here. You may hate kinetics. You may despise kinetics. I'm telling you right now, your body loves kinetics. Your body loves kinetics. It appreciates kinetics. It manipulates kinetics all the time. You must understand kinetics. It is the basis of life. We'll talk about later on. What life is about is kinetics here. All right, now, so again, you don't fear kinetics. Don't fear it. Understand it. Give me a chance to explain it next week or whenever. But don't worry about it. I'll got your back. I'll explain what it means as illustrated right here. All right, now notice here. So now what you're looking at here is, so once we get to kinetics, you'll start to understand why it's so important. So notice here, what you're looking at in this example is the first of three ways. The first of three ways the body chooses to turn on or turn off a pathway in metabolism. The first of three ways. And this way here is at the gene level. And any pathway being turned on at the gene level is a very slow way of turning on the pathway. It takes hours to turn on a pathway at the gene level. So why does it take hours to turn on a pathway at the gene level? And here it is. Because if you know molecular biology, it starts to make sense. Why does it take hours to turn on a pathway at the gene level? It takes hours to secrete the hormone, and part of the hours, it takes hours for the receptor to be found and to turn on the receptor. And part of the hours where then you have to go into the nucleus to find the enhancer region, fingering the DNA. And part of the hours, it takes time for the RNA polymerase to come in and then transcribe the gene into a primary transcript. And part of the hours, it takes time for the RNA to be capped, spliced, and tailed. That was chapter 3. It takes time for all that, and then it takes time for the messenger RNA to leave the nucleus to go onto the cytoplasm. It takes time for the ribosomes to jump onto the messenger RNA to start making protein. It takes time for the enzyme to be made. It takes time for the substrate to be made into the product. And all that time adds up to hours. That's why any pathway, this is the first of 3, ways to turn on and turn off a pathway. Any pathway being turned on at the gene level is a very slow process. So, part of the bigger picture of life, integration with endocrinology, is all the lipid hormones. They work at the gene level, therefore all lipid hormones work very slowly. Again, all that stuff that Dr. Wilson, try to integrate that stuff that Dr. Wilson talked about with this biochemistry here. All lipid, testosterone works slowly, estrogen works slowly, aldosterone works slowly. So any pathway being turned on at that gene level is a very slow way to turn on a pathway. Now the other 2 ways to turn on a pathway, stay tuned, coming up later. Don't worry about the other 2 right now, but this is the first of 3. At the gene level. All right, now what's coming up here is, now you understand this. Again, part of the big picture. You got to understand what's going on in the human body. Okay, now what's coming up next is, this is biochemistry integrated with molecular physiology. Here comes pathology. Now you can memorize diseases. You can memorize diseases up the wazoo. Just memorize this disease, this clinical manifestation, blah, blah, blah. Now I'm going to show you, illustrate to you, if you know a little bit about chemistry, how it goes a long way. Understanding the pathology with the biochemistry. If you know a little bit about chemistry. Here comes the question. What's the name of the disease in which these individuals secrete too much cortisol? Everybody will get this answer. Everybody will get this answer. What's the name of the disease in which too much cortisol is being secreted? What's the name of the disease? That's right. It's Cushing's syndrome. So Cushing's syndrome. Now, Cushing's syndrome, it's a disease in which too much cortisol is being released. Now let me show you how the biochemistry explains a lot of what's going on in Cushing's. And then after we're done, you'll say, oh yeah, now I understand that disease. Okay, now the idea here is don't worry about what these people look like. Don't worry about what they look like. But in terms of the laboratory values, what is the hallmark of Cushing's syndrome in terms of laboratory values? What's the hallmark of it? What's the hallmark of Cushing's in terms of laboratory value? Yeah, they have increased cortisol, but what's the laboratory value there? There we go, Daniel, that's right. And Rouge, that's right. Again, if I mispronounce your name, I apologize. I'm not very good at that. Lewis, that's right. And the answer is they have a severe hyperglycemia, a lot of glucose in the blood. Now, you can memorize that clinical manifestation, but how do you explain it? And how you explain it, you're looking at it. Right there on page 78. You're looking at what's causing the hyperglycemia. Right there. It's not hard. It's not hard. When you secrete cortisol, too much cortisol, therefore too much expression of the PEP carboxykinase in gluconeogenesis, too much gluconeogenesis, therefore too much glucose in the blood. There it is. Very simple biochemical explanation that explains the hyperglycemia. All right, now, what's another hallmark? I saw it earlier. I forgot who mentioned it. What is another clinical manifestation? What's another clinical manifestation of Cushing's syndrome in terms of laboratory values? I saw it earlier. I don't see it now. That's right, myelin. That's right. They have hyperlipidemia. That's right. They have hyperglycemia and hyperlipidemia. So how do you explain that without memorizing? Yeah, you can memorize. Maybe you can go through some mnemonic H and H, whatever. But I don't know what the mnemonic would be. But in any case, how do you explain it? And it starts to make sense. Here it is. Here's the explanation. When there's too much cortisol, you're going to generate too much glucose, hyperglycemia. How does the body respond to the hyperglycemia? I keep telling you, the body cares how much glucose is in the blood. So when the blood glucose rises, how does the body respond? What hormone is being released in response to the hyperglycemia? That's right, it's insulin. There we go. So insulin now becomes secreted, even though there's cortisol around, insulin now becomes secreted. And then insulin will come to certain tissues and tell those tissues, go turn on fatty acid synthesis, turn on triglyceride synthesis. You start making VLDL, all these type of things, and in the blood is tremendous amounts of triglycerides and VLDL. There it is, hyperlipidemia. It makes sense. Now, a lot of these triglycerides end up in adipose tissue in the back of the neck, giving rise to the clinical manifestation called the buffalo hump. And then in addition, a lot of these triglycerides get into the visceral adipocytes, giving rise to the purple stria. That's diagnostic for Cushing syndrome. But notice how the biochemistry explains all that kind of information. It just makes sense. Don't go memorizing. You should want to know this information. Not memorize it, you should want to know what goes on here. All right, so we're done now with chapter 5. You see how important it is. And later on in metabolism, if I say to you on individual metabolic pathways, if I say this pathway is being turned on at the gene level, just refer back to page 78. So whatever the hormone is turning on at the gene level, it will come. Now you go back to page 78. So from time to time, I'll say we already talked about how you're turning on this particular pathway conceptually at the gene level. All you got to do is go back to page 78. And I'll say, how do you know this is being turned on at the gene level? And you're going to use the public chat pod. You're going to put P78. Master this figure. Don't go memorizing it. Master it. Verbalize it. Use your words to explain. How does cortisol turn on glucose production in the blood? Just without the details, just explain it. It'll come to you eventually. Oh yeah, now I understand. Makes sense out of it. Okay, all right. Now what's coming up next? Gehane, you say that now you're starting to believe that biochemistry is the most fundamental science. I have to like it. Let me say something related to what you just said, Gehane. Is all these instructors who come into doing the reviews. Every instructor likely is going to say, every instructor is going to say, like Dr. Wilson probably says, physiology is the most important discipline. And then Dr. Barone is going to say pathology is the most important discipline for the boards. And Dr. Harris, pharmacology is the most important discipline for the boards. The 3 Ps. Physiology, pathology, and pharmacology. Then all the other people, Dr. Muscatel, the microimmunology, the most important disciplines. I'm not going to say that about biochemistry. I am not going to say that about biochemistry is the most important discipline. I'm not going to say it. But if I were to say something about biochemistry, is that if you don't know biochemistry, you don't know Jack. Okay? Now, don't ask me who Jack is, okay? But if you don't know biochemistry, you don't know Jack. There's no need for me to tell you that biochemistry is the most important discipline. I'm not going to say that. But if you don't know it, you don't know Jack. Who is Jack? Iron? That's right. Watch some movies sometime. Go to the movies and you'll see guys like Harrison Ford. They'll talk about Jack. You don't know Jack, There's another name. There's a surname to the Jack that I want to talk about right now. All right, so now what's coming up next is we're done with chapter 5. And now we're getting into chapter 6, and then after that, chapter 7 here. So now all that molecular biology that we just discussed, all these terms like replication, transcription, translation, and regulation of gene expression, the fundamental molecular biology. Now chapter 6 and 7 is now how you manipulate DNA from your chromosomes and so on. Now this first, in chapter 6, deals with recombinant DNA. Now first of all, definition. What does it mean? Recombinant DNA means the splicing together of two kinds of DNA. We take two pieces of DNA. One piece of DNA can be human DNA, and the other piece of DNA can be bacterial DNA. And by splicing them together, that's called recombinant DNA. On boards, if they talk about recombinant DNA, usually it's some human gene DNA and some bacterial plasmid. You splice them together, that's called recombinant DNA. So that is what the name means. Now, in this chapter is that, as I mentioned earlier on, that when you want to manipulate chromosomal DNA, because chromosomal DNA is too large, DNA cannot be manipulated very easily because it's too large. And so therefore DNA must be fragmented. And that's the first figure we're going to be talking about here, is how do you fragment chromosomal DNA? How do you break the DNA? And then the pieces of the DNA, how are they analyzed? And that's what chapter 7 is about, is how you analyze the fragmented DNA. Okay, so now the idea is fragmentation. Fragmentation. So again, recombinant DNA is a splicing of DNA from two sources. Again, most often it's some human gene. On boards, it would be human gene with some bacterial vector, but it doesn't have to be. It could be some chimpanzee DNA with some yeast vector. But in any case, it's recombinant DNA. Okay, now fragmentation. Now the idea is that if I were to do a biopsy of any tissue in my body, if I were to do a brain biopsy of me right now and take that biopsy and put it into a test tube, and then in the test tube are 46 chromosomes that come from me. And then in that test tube, if I were to add a particular detergent, what's going to happen is the detergent will now get rid of all the histones and all the proteins from the DNA. So all the histones will dissolve away. And then in the test tube, we have now negative DNA. So we have now negative DNA from 46 chromosomes. Now, don't go asking Dr. Mahad and Dr. Donald, what's the detergent you use, or what's the buffer you use, or what's the pH? Who cares? Let PhD students worry about that. For a medical student, all you got to know is you do a biopsy, you put in a test tube, you add some sort of detergent, and all the histones go away. And now you have negative DNA in a test tube. Now, in the test tube, then, we have 46 chromosomal DNAs that are now linear DNAs are stretched out. And in the DNAs, there are sequences of DNAs collectively called palindromes. Now I'm going to give you two definitions of palindromes, one now, one on the next figure. The first of the two definitions of a palindrome is it's a DNA sequence which is cleaved by a particular restriction endonucleases. There are many endonucleases, and each endonuclease looks for a different particular DNA sequence, and all those DNA sequences collectively are called palindromes here. Now, suppose there is a palindrome. Suppose this represents, what you're looking at here at the top of the figure, suppose this represents a portion of a very long chromosome that goes far in direction to your left and far in the direction to your right. This is a region of a very long chromosome. And suppose in this chromosome there is a gene shown in the orange. Just upstream is the promoter, here's spacer DNA, and upstream there's some enhancer region. Okay, nothing new compared to what we talked about earlier. Now suppose in the region of this gene there are several palindromes. The same palindrome, but it's in different locations. Suppose, as shown by the vertical line, the gray line, suppose as shown by this arrow, there's a palindrome here, even within the gene, here and here. Now let me give you an example of a particular palindrome. An example of a palindrome we're going to use in this chapter, chapter 6, as well as in chapter 7. The same palindrome will be used, and that example is GAA-TTC. Suppose that sequence, GAA-TTC, is found here, found within the gene. GAA is a codon, TTC is also a codon, but so it's found in the gene. And suppose the palindrome is found here and also found over here. All right, so this palindrome in the vicinity of the gene is found in 4 different places. Now, the same palindrome is also found in the rest of the chromosomes, hundreds of times, if not thousands of times. I mean, how often you remember, 23 chromosomes have 3 billion nucleotides. How often do you expect to see the sequence GAATTC? In 23 chromosomes, it might be found 100 times, if not thousands of times. But in the vicinity of this gene, it's found in these 4 regions. Okay, so my chromosome of DNA is sitting in the test tube. Now, in this test tube, if I were to add a particular endonuclease that looks for GAA and cleaves GAATTC, that's the palindrome for a particular endonuclease. Now, the name of this endonuclease that looks for GAATTC is called EcoR1. So EcoR1 is an example of an endonuclease, and its palindrome is GAATTC. And suppose then it's found at these regions, and then in the test tube, if we were then to add the endonuclease EcoR1, it's going to cut the DNA here, even within the gene, fragmenting even the gene. It cut here and cut there. Notice in the vicinity of the gene, we have a large fragment, a medium-sized fragment, and a small fragment in the vicinity of the gene. Now, in that test tube, how many fragments are we going to get? Hundreds of fragments, if not thousands of fragments, coming from 46 chromosomes. Fragmentation of the chromosomal DNA into pieces. And in this mixture of DNA pieces, some of them these fragments will be large, some will be small, some will be intermediate size. All kind of fragmentation of the chromosomal DNA. All right. Now, what's coming up next is some more information about the endonuclease issue as well as the palindrome. Okay, now on the next figure is, before we talk about an endonuclease, let me compare it with an exonuclease, because we talked about exonucleases earlier. Now, what does an exonuclease do? By definition, an exonuclease begins at one end of nucleic acid and cleaves one nucleotide at a time, just like a Pac-Man, removing one bead at a time from a chain of beads. Pac-Man just cleaves off one at a time. Matter of fact, in this course, we talked about 2 important exonucleases. Here it comes. You know I'm going to ask you, so here it comes. Without looking in the book, there was an example of an enzyme called the 5' exonuclease. What did that enzyme do? Let me see your answers. What did the 5' exonuclease do? What did the 5' exonuclease do? It removes the primer. That's right, the RNA primer. So again, the 5' exonuclease was a very important exonuclease. And then we also talked about a 3' exonuclease. What did the 3' exonuclease do? That's right, it's involved in proofreading. That's right, when DNA polymerase pauses and checks, the 3' exonuclease activity that the enzyme has will then cleave off a mispaired nucleotide. So again, 2 exonucleases that we have already discussed. Now, to compare it with endonucleases, by definition, they cleave internally in DNA. They cleave internal phosphodiester bonds, and they fragment the DNA, generating these things called restriction fragments. They don't, I'm going to stress, they don't cleave randomly. They cleave only at certain regions called the palindrome here. Now, let me stress here as well that there are about 200 different kinds of endonucleases that are commercially available. Matter of fact, people won years ago, they won the Nobel Prize for their discovery, because endonucleases are very powerful tools that are used in molecular medicine, as you see here in chapter 6 as well as in chapter 7 in genetic testing. So endonucleases are widely used. The National Board expects you to understand the concept of what an endonuclease does. So there are about 200 different endonucleases that are commercially available, and each one cleaves a different palindrome. Depends what the sequence is here. We know what these endonucleases are. We know what their palindromes are as well. Now, they're commercially available. You can go to Walmart right now. You can probably buy Eco R1. I don't know. They're commercially available. No big deal. But when they were first discovered, that was a big deal because they were very powerful tools. But nowadays, endonucleases, everybody understands what they do. Very powerful tools. All right, now, so these endonucleases cleave the DNA only in a region collectively called the palindrome. So here is the second definition. So we'll do this figure, then we'll take the break. A palindrome it's a DNA sequence which reads the same backwards and forwards. And usually a palindrome is 4 or 6 base pairs, 4 or 6 nucleotides in length. It doesn't have to be, but for the boards, a palindrome will either be 4 or 6 base pairs in length. Now, here's an example of the palindrome we talked about in the previous page. 5' GAA TTC to the 3' end. And that's the palindrome for ECO R1. Now you're going to look at that sequence and say, hey, that doesn't read the same backwards or forwards like the word Xanax, Xanax. That reads the same backwards or forwards. But in a palindrome, in DNA, it reads the same backwards or forwards on the opposite strands of the DNA. Now, notice here, upper strand, 5' GAA TTC to the 3' end. On the opposite strand, it reads the same. 5' GAA TTC to the 3' end. So on the opposite strands, starting with the 5' end, it reads the same. And that's the palindrome in this example for ECO R1. And then as a result, ECO R1 will cut the palindrome between the G and the A here, and the G and the A there. Here. Okay, now the idea here is that you can imagine, now is there an easy way to determine if a sequence of DNA, double-stranded DNA, excuse me, is there an easy way to determine if a sequence of double-stranded DNA, is it a palindrome? Is there an easy way to determine this? And the answer is yes, there's an easy way. Because you can imagine how difficult it would be sitting there on a computer monitor in front of you during the board exams, figuring out, does it read the same backwards or forwards? Or on board exams, they might give you one strand. And you say, is that a palindrome? And then you draw the other strand. Man, it takes a lot of time. Is there an easier way to determine if a sequence is a palindrome? And the answer is yes. All you got to do is look at one of the 2 strands of double-stranded DNA. Just look at one of the 2 strands, like you see right here. One of the 2 strands of double-stranded DNA. And in your mind, bend it in half. Use my trusty cord again. Here is now one strand of double-stranded DNA. And in your mind, bend it in half. And when you bend it in half, ask yourself the question, does the base on one side base pair with the one on the opposite side? If the answer is yes, you go to the second nucleotide. Does that one base on one side base pair with the one opposite side? If the answer is yes, you go to the third nucleotide. Does that one nucleotide on one side base pair with the one on the opposite side? If the answer is yes, it's a palindrome. Here it is. Look at this sequence here. In your mind, bend it in half. Notice how the A base pairs with the T, A base pairs with a T, and the G base pairs with a C. And this is the palindrome for ECO R1. Very simple way to determine if a sequence is a palindrome. Not hard at all. Okay, now what we're going to do is we're going to take a 10-minute break. Okay, and then we're going to come back, and then we're going to get into a comment or two about cloning, and then we're going to move on. Okay, so let's take a 10-minute break, and I'll see you shortly. Okay, everybody, welcome back for hour three. So we just talked about cutting up a DNA with endonucleases. So in medicine, then why do you want to cut up DNA, chromosomal DNA? I mean, if what's the purpose of cutting up the DNA, chromosomal DNA? And there's two reasons that you want to cut up DNA. And one reason is for cloning DNA. That's what chapter 6 is about. And the second reason is for analyzing the DNA, genetic testing for diseases and so on, and forensics. So again, why you want to cut up the DNA? For cloning purposes and for analysis purposes, genetic testing. Okay, now chapter 6 deals with cloning. Now let me say something about this, cloning business. Now coming up, there's like in your book, there's like 5 or 6 or 7 pages that talk about the methodology of cloning. Now, remember earlier I mentioned to you that I go against the grain. Most educators will tell you it's best in terms of studying. It's best for you to read ahead, to learn, understand what the instructor is going to talk about. And I told you I go against the grain. My advice is not to read ahead, because you might be studying something that I'm going to come in and says it's low yield. And here comes the example of this, one example, because we're not going to talk about the methodology of cloning. It's in your book, the details of cDNAs, it's all in your book. Low yield stuff. Let PhDs worry about that kind of stuff. But for doctors like you are, or going to be a doctor, it's what you're going to do with the clone gene. Again, cloning means you take a little bit of DNA and you make more DNA. And for reasons will come up. So the idea is, so the methodology, how you splice the DNA into a bacterial vector and all these other things, and how you it's in your book. If you want to know the process, help yourself. I'm not stopping you. But it's chances of you seeing that on board exams, the methodology of cloning, the chances of seeing it are low. Take a chance that you won't see it. Focus your attention on high yield items. We just talked about Cushing syndrome and cortisol, zinc finger receptors. We talked about all that stuff with the PPARs. Those are high yield. That's what you need to focus your attention on. You can't know everything, unless you're a type A personality, you want to know everything. But the idea is focus your attention on what are high yield items. So the methodology of cloning would not be concerned. So now the issue is, well then what would you do with a clone gene? So okay, suppose I clone one of my gene, and in a test tube I have millions and millions of copies of a gene that I made millions of that particular gene. Whatever the gene was, it was a single gene, and I made millions of copies of it by cloning methodologies. What would I do with that clone gene? Okay, there are two reasons why what you could do with a clone gene, and why you need to know this for medicine. And one reason is, one use of a clone gene, make millions of copies of it, is to splice it into a bacterial vector to make recombinant DNA, a human gene with a bacterial DNA, recombinant DNA, and then take that recombinant DNA and give it to E. coli or some other harmless organism. And as E. coli grows, that bacterial vector also is replicated along with the human gene. And so now that human gene is now being made as E. coli grows. As E. coli grows and grows and grows in some big vat, it's making large amounts of the particular gene, and then the gene can be expressed into protein. Again, the details are in the chapter, but I wouldn't worry about that. But the idea is now that gives rise to recombinant proteins. And in other words, these are human proteins that are made from bacteria. And so the idea is, so why the National Board expects you to understand this is the concept of what you could do with the clone gene is, because a lot of the drugs that you will administer in medicine, a lot of these drugs are generated by recombinant DNA technology. For example, insulin. And in the old days, old being 15, 20 years ago or something like that, when somebody had type 1 diabetes and you administered, if you're a physician then, you would administer insulin. Where did the insulin come from? Let me see your answer here. Where did the insulin come from? That's right. It's right. That's right. It usually came from pigs. You had to sacrifice a half a dozen pigs or something like that to get enough insulin to treat type 1 diabetes. And that was porcine insulin versus human insulin, so you might have problems along that line. But the idea is you got it from animals. But nowadays, when you administer insulin to treat type 1 diabetes, where does the insulin come from? It's human insulin. And it comes from recombinant DNA technology. Some companies out there, they clone the insulin gene from a human being. The E. coli now replicates it, makes large amounts of the insulin gene from me and you, and expresses it into insulin protein. There it is. Other examples, the blood clotting factors that treat the hemophilias are generated by recombinant DNA technology. Factor VIII, for example, comes from recombinant DNA technology. A lot of vaccines, as illustrated here by the hepatitis B surface antigen, generated by recombinant DNA technology. So a lot of the drugs, matter of fact, 1/4 of all new drugs that are being generated pharmacologically by pharmaceutical companies, 1/4 of all new drugs are proteins, the protein-based drugs, and they are, again, generated by recombinant DNA technology. Other examples, EPO, erythropoietin in treatment of blood disorders, growth factor in the treatment of dwarfism. These are proteins that are generated by recombinant DNA technology. Again, don't worry about the details, but understand why you want to clone a gene in the first place. Now, the other reason, the second reason that you want to clone a gene, I'm going to spend a little bit of time talking about, is that's up and coming in your profession. Up and coming in your profession. And that is gene therapy. Gene therapy. Where this is right now, gene therapy in medicine is just in its infancy, and it was started about 10 to 12 years ago, but still in its infancy. Where what you're trying to do is take a human gene that's normal, a therapeutic gene that's normal, and give it to somebody who has a mutation in that particular gene to cure a particular disease. So what's going to come up is two figures. The first figure is the principle of retrovirus gene therapy, the principle of it, and then after that, the actual application of retrovirus gene therapy. Now, there are not that many diseases currently that are treatable by gene therapy techniques, because right now it's in its infancy, and on the next figure we'll talk about why is this type of therapy not progressing rapidly. So we'll get to that issue in a few minutes. But the idea here is again, this is the principle of retrovirus gene therapy. Now, suppose there is a human being that has a genetic disorder. They may have a missense mutation of a particular gene, nonsense mutation if they survived, all the kind of stuff we talked about yesterday. In any case, there is a gene that's mutated that's causing pathology in that individual. So we're going to try to replace the defective gene with the good gene. So what's done is to take some retrovirus. Now, you just had Dr. Muscatello in virology, and she talked about the retroviruses. And remember, the retroviruses have several genes. For example, they have the gene GAG gene, the Paul gene, and the envelope gene, a few others as well. But suppose they have these three genes. Okay, now what's done is in a test tube, we take that retrovirus, and then as shown by the arrow, we remove a portion of the retrovirus, just get rid of it. And when we get rid of that portion of the retrovirus, we then add into the virus, as shown in the orange rectangle, we then add the therapeutic human gene. Where did that gene come from? Cloning technology. So you make millions of copies of a particular gene in a test tube. Now I got large amounts of this gene. And so now I can take a little bit of that gene and put it into the retrovirus. And now you might say, hey, I thought retroviruses are RNA and genes are DNA. Yes, But nowadays, you can go backwards and forwards, DNA to RNA, RNA to DNA, back and forth, no big deal. We got enzymes to do that. Don't worry about that level of detail. It's simply we can take a cloned gene, DNA, make it into RNA, and then put that RNA into the retrovirus. Here, okay, now so now notice what we got is a recombinant virus. So here is the virus in the blue and the yellow there, and in that orange rectangle represents the human gene. Actually, technically, it's RNA. So this is a recombinant virus. But importantly, this recombinant virus cannot replicate itself. In other words, when the virus is given to a human being, it will not cause pathology. It will not cause pathology. And the reason is the portion of the virus that was removed was responsible for enabling the virus to replicate. By removing that part of the virus that causes the virus to replicate, then the virus cannot replicate. And now we substitute into the virus the human gene. But now the virus is faked out. The virus thinks that new human gene is part of its own genome. So the inserted human gene is part of the viral genome. The virus is now faked out. It says, oh, look at me, man. Look, I got some, look at me. I got some human gene in me. I am nice and pretty. Because I got some human gene, man. I am nice and good. I'm good to go. So anyway, the virus is faked out. Okay, don't tell the virus, but the virus is faked out because now it has a human gene in it. So then what's done is to take that RNA of the virus and package it with membranes, with lipids to make membranes to make it look like a retrovirus. And again, don't worry about the detail. Don't worry about that. It's the concepts. So now the RNA of the virus looks like a virus because it's packaged in membranes. Okay, so now that virion is ready to go. So then we take some, excuse me, we take some human target cell. And suppose in this target cell, here is in the purple represents the nucleus. And this is one of the chromosomes in the nucleus. And suppose in this chromosome, there is a defective gene that we're trying to replace with a therapeutic gene. Okay, so then we take this target cell, we give the virus the replication defective retrovirus, the recombinant virus. We then give it to the human target cell. And then, as you know from virology, the virus now will attach to the plasma membrane, and then the RNA of the virus comes in, and then reverse transcriptase will take the RNA of the virus and make it into DNA. And then there's that enzyme that will now integrate the DNA into the chromosomes of that target cell. Uh-oh. What's the name of the enzyme? Help me out here. Oh, it's on the tip of my tongue. The name of the enzyme that integrates the DNA into the chromosomes in virology. Name of the enzyme that integrates the DNA into the chromosomes. Oh, that's right. Yeah, that's right. Wow, that's right. That's right. It's called integrase. That's right. I know. Sometimes I get a mental block in biochemistry. Sometimes, That's right. Integrase integrates the virus into the chromosomes of the host cell. Okay, but now importantly, again, the virus cannot replicate itself. The virus will not leave this cell and invade another cell because it can't replicate. But what the virus can do, it can then express its genes into proteins. And one of its genes is the therapeutic gene. There it is in the orange. That's a therapeutic gene made into the therapeutic protein. There it is right there, I should say. The gene product, the protein. So the therapeutic gene made into RNA, made into protein, which you see right here. And then the protein is now curing the cell of the problem, whatever the problem happened to be. Okay, so this is now retrovirus gene therapy. Yes, I know I oversimplified it. Yes, I get it. I'm trying to get you to understand. You'll never understand details unless you understand the simple stuff, the concepts. Make sense out of it. Make sense. All right. So now is the issue of how is this type of technology used in your profession? Okay. So this is the example I'm going to choose here. This is also integrates with immunology here, as well as pathology. Now, suppose this little child here, but this is, by the way, this is called ex vivo gene replacement therapy. Now, first of all, the words ex vivo, what ex vivo means. It means you take cells out of an individual, you genetically modify those cells, and then you give those cells back to the same individual. Take cells out, genetically modify, and give them back to the same person. That's what ex vivo means. Now, suppose that this child right here in pathology and immunology has one of the severe combined immunodeficiencies. Because this patient, so he has one of the SCIDs, because this patient has a defective interleukin receptor gamma chain gene. So the gene is mutated, and therefore interleukin receptor gamma chain protein is abnormal, causing SCIDs in this child. Okay, so now we're going to try to replace the defective gene with the good gene. So what's done, and as you all know from Dr. Muscatello's lectures, the protein, interleukin receptor gamma chain protein, needs to be expressed in CD34 plus bone marrow cells. So what's done is to take out do a bone marrow aspiration and take out the CD34 plus bone marrow cells, purify them, and take those cells and place them in a plate or some flask. And while they're in this flask or dish, they're kept alive with growth factors. This is kept alive. While that's being done, we take a replication defective retrovirus from the previous page, and then we add the therapeutic gene, interleukin receptor gamma chain gene, into the retrovirus. Package it with membranes to make it look like a virus, and then give those viruses to the CD34 plus bone marrow cells, and now the virus comes into those cells, and because it's carrying the interleukin receptor gamma chain gene, that DNA of the virus now incorporates into the chromosomes of the CD34 plus bone marrow cells, and it'll integrate, and then the interleukin receptor gamma chain protein is expressed, and therefore curing these cells of the problem. After that genetic manipulation, these cells are then transferred back into the same individual, and in quotation here, curing a defective tissue here. So again, now this person now has the good gene and now making good protein, curing the pathology in this individual. Now, as you might imagine, these CD34 plus bone marrow cells will eventually start dying out. And so how often do you have to do this type of therapy, whether it's 1 month or once a year? It doesn't matter. Don't worry about that level of detail. Just understand the concept, how often you have to do this type of therapy, because those cells will die out likely. And so the idea is that this probably would have to be done throughout the child's lifetime. Now, in your profession, this type of therapy is only being done for a handful of diseases. Several examples of this is in SCID. One of the other SCIDs is ADA deficiency. The most common SCIDs, adenosine deaminase deficiency. In fact, that was the first successful replacement of a defective gene with a good gene, the ADA gene. So that was the first example of success with this type of therapy. It's also used for the other SCIDs we just talked about. Another example where this is being done, or was tried and done, was the hyperammonemias. Now there is, matter of fact, you might have first heard about this technology about 12 or 15 years ago at the University of Pennsylvania in Philadelphia. There was, when this technology was just starting out, there was a man, a 21 or 20-year-old man in Philadelphia that had hyperammonemia because the OTC gene, OTC stands for ornithine transcarbamylase. We'll talk about this enzyme a little bit later in the urea cycle. So he had a urea cycle defect, and the urea cycle was located in the liver. And so what happened was, at Penn, the physicians there cloned a therapeutic OTC gene from human, and then they took the OTC gene that's normal and therapeutic, and they put it into a replication defective adenovirus. Why adenovirus? Because the adenovirus should invade the liver cell and bring the gene OTC into the liver cell, because that's where the urea cycle is located. But unfortunately for that man, he died a day or two later. And then what happened was, it was written up in the newspapers and magazines, and it set back gene therapy very severely because they were, again, this is new, this is like a novel science fiction thing they were trying on humans, this genetic manipulation, and it didn't work. But unfortunately for that individual who died, these are technical problems. Now, as I mentioned a few minutes ago, this type of gene therapy is only in its infancy. Even though it's like 10 or 15 years of age, this type of therapy, it's not advancing very rapidly. Now, why is that? Let me see. Now the reason why it's not advancing very well, because there's major problems with this type of therapy. Let me see your answers here. What's money? Besides money, that's true. Money would be a problem. Always money. But in terms of molecular issues, what do you think a major problem with this type of therapy is? Now, let me ask you this. Besides immunological problems is a problem, but it's not a major problem. These other more major problems. But here comes one of the major problems, and that is, why are viruses being used here for these with the gene therapy? Retroviruses for certain diseases, and adenovirus for the OTC deficiency. Why are viruses being used? Why are viruses being used? And the answer is to integrate and express properly. Well, that's true, but even more simple than that is this: because the virus is targeting the right tissue. You got to take the gene and put it into the right cell, the right cell type, the right tissue. Is because retroviruses invade white cells. And SCIDs, the white cells, are genetically defective. That's why you use retroviruses to treat SCIDs. Whereas in OTC deficiency, the urea cycle, adenoviruses invade the liver. So the viruses are being used as a delivery vehicle to deliver the gene into the right targeting the right tissue. Here, if the gene is deposited in some other tissue where the gene doesn't belong to be expressed, that's a problem. In fact, that's probably what happened in that man who had the hyperammonemia. I think I read, I could be wrong, I think I read where that the adenovirus invaded some other cell type and deposited the gene into some other cell. It was being expressed, it doesn't belong in the other cell, and it caused the death in that individual because it messes up metabolism. All right, but so targeting the right, now you might ask, well, why don't you just take the gene and take it orally, a therapeutic gene, and take it orally? What's the problem with that? Why don't you do that? Why don't you might think, why not take a therapeutic gene and just take it orally? That's right, it would never survive digestion. And you say, well, okay, then why don't you just take the gene and IV it into the bloodstream? Let me see your answers here. Why don't you just simply IV the therapeutic gene into the bloodstream of an individual? Why not? That's right, Gisela, that's right, because there's this thing called the plasma membrane, and genes are negatively charged. Remember all those phosphodiester bonds? Genes are negatively charged. They would never cross the plasma membrane. They would never get into the cell. They would stay in the blood and eventually peed out. They would never enter a cell. So that's why viruses are being used. They are a delivery vehicle. Now, suppose that you did use the right gene, the right virus, and suppose it did come into the cell, come into the right target tissue. What's another major problem with this type of therapy? Suppose we got the gene into the right tissue by the virus. What is another major problem with this? That's right. There we go. And that is to integrate correctly. By the way, some of you said, hi, Zia, the immune response. No. The immune response would be if the protein is out there in the bloodstream. If it's inside the cell, there's no immune response per se. So the idea is that once the gene comes into the cell, the major issue is it must integrate correctly. But the problem is the gene might integrate incorrectly. And if it integrates incorrectly, that can be a problem. As an example, where one gene integrates incorrectly with another gene. In other words, you can have two genes, the therapeutic gene integrating with a normal gene that sits in the chromosome. They can integrate side by side. And that's a problem. Can you think in pathology? What's the name of the disease in which one gene integrates incorrectly side by side with another gene? Anybody know that disease? What's the name of the disease? Anybody? Where one gene integrates incorrectly with another gene side by side. Okay, and okay, now if I were to ask, nobody got the answer yet. If I were to ask you, what's the name of the chromosome called? What's the chromosome? That's the Philadelphia chromosome. Remember CML, chronic myelogenous leukemia. Remember all that? Some of you said translocation. That's true. It's reciprocal translocation. We'll talk about tomorrow. But the idea here is that when you get translocation, where one gene, the BCR gene, now fuses side by side with the BCR gene and the ABL gene fuse side by side. That's called the Philadelphia chromosome. And now you're getting expression, abnormal expression of the proteins being made. So it must integrate correctly. All right, now let me ask you this. Suppose another major problem. Suppose the virus gets the gene into the right cell. And suppose the gene integrates correctly. No problem with fusion with other protein, other genes and so on. Integrates correctly. What do you think another problem might be? Major problem. That's right. Expressing it, that's right. Is you have to regulate the amounts of the expression. If the gene never gets expressed into RNA, it's useless. But if the gene gets expressed into too many enzymes, that's going to mess up the cell, right? So, so the idea is it has to be regulated. How much gene is being regulated into the enzyme. So again, notice these major problems. Again, there are other problems that could arise, immunological problems that could arise, the HLA and all that kind of thing. But the idea is that's why this type of technology is only in its infancy. But it's just a matter of time. 10 years from now, 15 years from now, 20 years from now, you may be doing this with your own finger. You may be doing gene therapy in your own office, your own clinic. You may be doing this with your own fingers. 20 years from now, 10 years from now, I'm going to be dead. I'll be 6 feet under. And you'll be walking over my grave, and I'll be saying to you, hey, Dr. So-and-so, remember me? I taught you this. Remember on May 7th, 2014, I taught you all this. Remember, don't walk over me. Throw me a flower. Oh, no, don't walk away. Yeah, because it's the matter of time before this becomes much more commonplace. These are technical issues that must be surmounted. Now, let me put this into perspective as well. Years ago, when I started graduate school, no one ever talked about cloning of genes. It was like science fiction. No one ever talked about clone. How you clone a gene? It was science fiction stuff. Nobody ever thought of that when I started graduate school. And then in the 1980s, somebody came up with the endonucleases that cut up DNA, and then shortly thereafter, wasn't that far afterwards, where the first gene was cloned, and it made the headlines. You made Science magazine, Science journal, and all this kind of stuff. It was headline stuff, the cloning of the first gene. But then after that, it became fairly commonplace. And nowadays, we're in 2014, nowadays, you say you clone a gene, and somebody will just yawn. Oh, so what? It's just commonplace stuff. That's the same thing with gene therapy. Right now, it's only in its infancy. It's make headlines when it works, but eventually it'll be a much more commonplace type of technology. Okay, now another item here. Yeah, now, by the way, Dolly the sheep, that's something different. No, that's cloning of chromosomes. That's that's altogether different. No. Animal cloning that's used in veterinary medicine is much different. What you're doing there is, if I could verbalize this, is what you're doing in animal cloning. You're not talking about a single gene. In animal cloning, you're talking about whole chromosomes. What's done is to take an egg from a female animal. You take the egg, which has 23 chromosomes. Well, if it's female, if it's a human, it'd be 23 chromosomes there. I have no idea how many chromosomes are in sheep or cattle. But in any case, you take out the chromosomes out of that egg. And now in the egg, there are no DNA, there's no chromosomes. But in that egg are all these enzymes that are waiting for fertilization. It's like a primordial environment. So the egg is sitting there with no DNA, no chromosomes, waiting to be fertilized. But the DNA, the chromosomes were removed. But then what's done is to take a somatic cell out of another animal, another sheep or whatever, and the somatic cell is diploid. Remember that? It's diploid and two sets of the chromosomes. And then you take that diploid set of chromosomes and you take them out of the somatic cell and put them into the egg. And now the egg got faked out. The egg thinks it just got fertilized because the chromosomes are now diploid. And so as a result, now those enzymes in the egg now start the replication process, and then you take that egg that now has a diploid set of chromosomes, you then implant into a foster mother, and then you wait the number of days or months, and eventually you end up with Dolly the sheep or something like that. But the concept is those are chromosomes, not genes here. Okay, so that is done in veterinary medicine. Okay, but now the next issue is now this type of technology is permitted in humans. Everybody listening to me right now in the audience, I don't think anybody would have any type of ethical or religious problem with this type of therapy, gene replacement therapy. I don't think anybody would have a ethical or religious problem with this type of therapy. And the reason why is because this is somatic cell gene replacement. That we're taking a somatic cell, putting in the good gene, and putting it back. Somatic cell gene replacement. And the important feature is, in terms of ethics and religion, is the gene is not inherited. Not inherited here. So that's why there's no problem with this type of situation that goes on. Okay, now the other item here is now if you do have an ethical problem with this type of therapy or a religious problem with this type of therapy, then you should have a problem with a discipline called pharmacology, right? Think about this. In pharmacology, suppose a patient has type 2A hyperlipidemia because they have a defective LDL receptor gene. So they have elevated cholesterol in the blood and all these xanthomas and all that. And so nowadays, how do you treat them? You treat them pharmacologically with a chemical, okay, and like statin drugs and so on. So you treat them with a chemical. But in the future, instead of giving them a chemical in pharmacology, why not give them the therapeutic LDL receptor gene that's normal, not mutated, to cure the patient? So that, again, it's not going to happen tomorrow, but somewhere in the future that might happen. So one more time, is that I don't think anybody has a ethical or religious problem with this type of therapy, because if you do, you should have an ethical issue with pharmacology, because pharmacology, you're just given a chemical to treat a particular disease. But the future of medicine is where you give the gene to cure a particular disease. Let me stress here, is the future of medicine is genetics, not only with treating diseases, but the future of medicine is genetics. It's going to be the future where you can, even in OBGYN, you think, well, OBGYN, you can, what's it got to do with genes? Well, you can do genetic testing and so on, genetic profiles of infants and things like that, of fetuses. So the future of medicine is genetics here. And you must understand the basic principles, why the boards hammer students on genetics here, molecular genetics here. Okay, that's the future of medicine here. Okay, now we're done. By the way, you should ask, well, if this can be done with somatic cells and is being done overtly in human beings with this somatic cell gene replacement, well, couldn't this also be done with germline single gene replacement? Yeah, and the answer is yes. That could be done. That could be done in the germline. But there you have all the ethical issues, all the ethical religious issues. This is where your profession is heading. You got to be alert that in your profession, as time goes on, there's going to be a lot of controversy that's going to come up regarding genetics. Now, what I mean by this is, as an example, suppose in the future, suppose some pregnant, you're an OBGYN, and some pregnant woman comes into your clinic and she says to you, well, I just had genetic profile done and I found out that I'm going to have a son. And I found out in my genetic profile that my son's going to have brown eyes. And I want my son to have blue eyes. So go to the shelf over there, Dr. So-and-so, and I want you to get the gene for blue eyes, put it into me, so my son can have blue eyes and look like Brad Pitt and star in trashy movies, wasting everybody's money. Yeah, that's right. That could be done. Yeah, so put the gene for blue eyes and you're fine. Or another example, suppose you're in OBGYN and some pregnant woman comes into your clinic and says, hey, I want you to go to the shelf over there and get the gene for growth factor and put it into me. So my son, I'm going to have a son, I want my son to be 7'1". So my son can then star in the National Basketball Association, making millions and millions of dollars, so I don't have to work the rest of my life. So put the gene for growth factor in me to make my son be 7'1". Now those are called designer genes. Designer genes that make people look different. And designer genes, I'm not talking about Calvin Klein genes. I'm talking about genes that make people look different. Now what I'm getting at here is there comes the controversy. I don't have the answer. All I'm saying is you better be alert to this, because as time goes on, it's going to be much more commonplace and it's going to be very controversial, where people are given genes to make their offspring look different from one another. Now you might say, man, I ain't going to do that because that is unethical to do that. That is against my religion to do that because you're playing God. That's unethical to do that. But then if it's unethical, and it is, in my opinion, but then what's the difference between that and plastic surgery or anything like Botox or something like that, where a person is looking different than what they were supposed to look like? There's where that fine line, that hazy line of what's ethical and what's not, what's permitted and what's not permitted. And so, so you got to be ready. This is where your profession is heading in the future. Okay. Okay, so now what's coming up next is, so we're done with this chapter. Now the rest of the chapter, by the way, comes to talk about transgenic animals. We're going to skip over that low yield item, how you make transgenic animals. What does it mean, low yield? I haven't heard about that much, maybe one time in the last 5 or 6 years. I wouldn't worry about transgenic animals. And I don't even remember if it was on the boards or if it was on MEs or something like that. Anyway, don't worry about knockout animals or transgenics. That's usually that PhDs worry about those things. Those are animals that have a genetic problem on purpose to study certain drugs, how certain drugs work and things like that. But for medicine, I would not worry about that. Okay, so we're going to skip over that. And now we're done with chapter 6. There it was. We're done with chapter 6. Okay, so you know what's important to know. Okay. Muscatella said it was important. What, transgenics? If she said it was important, I'm not going to disagree with her online here. I mean, if she says it's not important, it is important. All I can say is, let me give you the facts. I have heard about it being on the board only one time in the last 5 or 6 years. And even then, I don't know if it was a board exam or if it was an MBME. So if I were you, my opinion, I would not worry about it. Focus your attention on what gene therapy means, what we just talked about, high yield stuff. Now, Dr. Muscatello said it's important. Well, maybe take it, you could do it. No one's stopping you. Let me stress, no one is stopping you from looking at other things, But all I'm saying is you can't possibly know everything. You can't. You're not a flash drive and you put yourself into a computer and you come up with all this information. You have to use that what little information you have to use it to think. And that's what I'm trying to get across here. All right. All right. So anyway, so that's the idea. So now in chapter 7, what we're going to talk about is techniques of genetic analysis here. Techniques of genetic analysis. So what this is about is that when we want to analyze DNA or RNA or whatever, how does it happen? So you can imagine this is a high yield chapter because it is, again, modern molecular medicine. And it's now fairly widely used now, where you can look at certain genes for mutation and look at a pedigree, a family with a particular trait, and you can look at the actual chromosomes of an individual, look at the genes of an individual to see if they have the same mutation, something along that line. And you must know this chapter. All right, now let me say something here as well. Again, as I mentioned at the earlier in the first hour, is depending on where you went to medical school or where you went to medical school, you may not have talked about this stuff. Okay, because in fact, I talked to many medical students say, "Man, I never heard this stuff before," and so I don't do it. But again, I'll try to take it slow, but I can't go too slow because I don't want to bore people who know this stuff. So again, I'll try to take it at a pace which is understandable for people who don't never had this before, but at the same time not try to bore people. But again, don't worry about the detail. It's the concepts. Now, let me say something as well. It's the first figure. The first figure is the basis of the rest of the chapter. You must understand the first figure. So what we're going to do is, I look at the clock, it's 10 minutes to the hour. So what we're going to do is we're going to take a 10-minute break and then come back and talk about this all-important figure of blotting. You must understand blotting. If after I talk about it you don't understand it, you can forget the rest of the chapter because you're not going to understand the rest of the chapter unless you understand this first figure. So take a 10-minute break, come back, and come fresh because we're going to talk about this all-important figure. Okay, take a 10-minute break. See you shortly. Okay, everybody, welcome back to the fourth hour. So we're on the last chapter of genetic testing, and at the end of the chapter, I don't know if we'll get to it today, but at the end of the chapter, there will be a couple questions I do want to cover, vignettes based on stuff we've covered. Okay, so now again, you must understand this issue of blotting. It's conceptually not that difficult, but for some students, they get hung up on certain items, and I'll tell you what that is. I get so many questions on this, and you must pay attention, because I know what happens is that you're listening, this and that, and all of a sudden you get some students get distracted. They might get a text, or they get a phone call, or they get something to eat. They come back and say, "Where is this guy? Can you explain what he just said?" and this kind of stuff. And I don't mind explaining it as long as you're trying. You still don't understand it, but get rid of the distractions, because if you got to understand this first figure, because it's going to be the rest of the stuff depends on understanding what blotting is about. So the idea here is that there is, remember, when you want to analyze DNA for whatever reason, DNA is too large, and we talked about you got this series of enzymes called endonuclease that can cut up the DNA, generate DNA fragments, also called restriction fragments. And then the fragments are then analyzed. And as we talked about earlier, whereas RNA and protein are sufficiently small, they are not cut up when you want to analyze those proteins, and they are analyzed directly here. Like, for example, when you had immunology, you talk about the Western blot. That's a blotting technique. In the Western blot, you never talk about cutting up of HIV proteins. They are analyzed directly because they are sufficiently small. Now we're going to focus our attention. Now all of these substances are negatively charged, with one major exception. All DNA fragments are negatively charged because of all those phosphodiester bonds. All RNA are negatively charged because of the phosphodiester bonds. And most proteins, except histones, are negatively charged because they have a lot of aspartic acids, glutamates. They can be phosphorylated with phosphates on them and things like that. They have sugars on them that are negatively charged. But the idea is most proteins are negatively charged. The best example that's not are the histones. So when you want to analyze these things, then we can do electrophoresis here. Now the idea here is, now we're going to focus our attention on DNA, chromosomal DNA. Suppose I want to analyze my chromosomal DNA to determine genetically whether I am homozygous for sickle cell being normal. I'm homozygous normal for the beta globin gene. Am I a carrier for the sickle cell trait, or am I homozygous for the sickle beta globin protein? Okay, so I want to analyze my beta globin gene to determine my status. So again, so conceptually, what's done is to do a biopsy of any cell in the body. I don't have to go to the bone marrow. I can go to any cell in my body. I could just do skin biopsy or whatever, and put that biopsy in the test tube. And in the test tube, I add, in this example, I add some detergent to get rid of all the histones. And then to the chromosomal DNA, I add a particular endonuclease to cut up the DNA. And then, so in that test tube, from 4 to 6 chromosomes that came from my biopsy, there may be hundreds or thousands of fragments. And some of those fragments will have the beta globin gene fragments. Now, what I mean by this now, this is going to be coming up, is the difficult issue for a lot of students. Suppose, if I take my chromosomal DNA, fragment it with an endonuclease to generate hundreds, if not thousands, of fragments. So I got, and then some fragments are large, some are medium sized, some are very small, mixture of fragments. So I take that mixture and subject it to electrophoresis. So you put it on the top of the gel, and then they're going to separate. Because they're negatively charged, they're going to migrate to the positive terminus. Because all DNA fragments are negatively charged, they're going to migrate to the positive terminus by electrophoresis here. Okay, now let me ask you this. So the fragments then, as they migrate to the positive terminus, they're going to separate on the basis of what? Let me see your answer. They'll separate on the basis of what? Separate on the basis of size or length. That's right, size, not charge. They're all negatively charged. They're separating on the basis of size. Now let me ask you this. Will the largest fragments be at the top of the gel or at the bottom of the gel? If we take the mixture, apply it to the top of the gel, will the largest fragments be at the top or at the bottom? That's right, at the top. So the largest fragments are at the top and the smallest fragments are at the bottom. Now why? Because the small fragments, because they're negatively charged, they migrate to the gel. Remember, the gel is like jello. They migrate through the gel very quickly on the way to the positive terminus. They move very fast. Whereas the large fragments, they get slowed down, they get retarded, they get impeded by the gel matrix. So they have a hard time moving through the gel and on the way to the positive terminus. So therefore, the large fragments are at the top, the small fragments are closer to the bottom. Please keep that in mind. Okay, so they're separating on the basis of size only. Okay, so then what's done is to take that gel and then line it up side by side with a piece of paper. Let me take a piece of paper here. There's a piece of paper. Okay, so suppose this is the gel, and on this gel are all these bands that came from my chromosomal DNA, and I look at that gel and I see nothing. Why? Because DNA is colorless. But I know there's fragments from beta globin on this gel, but I can't see them with the naked eye. So what's done is take a piece of paper and line it up side by side with a piece of paper. Line it up side by side with a piece of paper. And now electrophoresis in the direction of the paper. So all of these bands of the gel are now transferred onto this piece of paper. So then, there it is, right there. So you transfer from the gel onto the piece of paper. So on this piece of paper, the large fragments are at the top and the smallest fragments are at the bottom. And after the transfer into the paper, I look at that paper and I see nothing. Why? Because DNA is colorless. But I know that the large fragments are somewhere and the small fragments are down here, and somewhere there is the beta globin gene fragments. So now the issue is, by the way, you might ask, why are you transferring the paper? Because did you ever work with Jello, manipulate Jello? You know, Jello falls apart. If you're going to try to manipulate the Jello to get to look at bands, gels fall apart. Whereas paper, this piece of paper, watch. It's still a piece of paper after you manipulate it. If I'm a slob in the lab, I gotta get the piece of paper. I don't know what's going on, but the piece of paper doesn't crumbles, but it's still there. So the gels are still there. The bands are still there. That's why you transfer the paper, because you can manipulate the paper much more easily than a piece of gel. Okay, so now on this piece of paper, there's the large fragments at the top and the small fragments are at the bottom. Okay, now what's next done is then the question is detection. How do I detect the beta globin gene fragments? Because after all, why am I doing this? I'm doing this to analyze my beta globin gene. Remember, I have two genes, one from my mother, one from my father. I'm analyzing my beta globin genes to see if I'm a carrier or homozygous for beta globin sickle cell. Okay, so now what I'm going to do is I'm going to digress from this piece of paper for a minute and explain something that a lot of students have a hard time, especially if you've never worked in a laboratory. This is where it becomes difficult. Now in the lower left quadrant here, suppose I have this black line is part of a very long chromosome that goes in both directions. Portion of a very long chromosome, and this black horizontal line represents the beta globin gene. Now suppose within the beta globin gene there are palindromes. These dotted lines represents palindromes for ECO-R1. Remember, the palindrome was GAA-TTC. Suppose within beta globin gene there's a palindrome GAA-TTC right here, right there, and right there. Now within the rest of the chromosomes and so on, it's present many, many times. But in the vicinity of beta globin, it's present at these four positions. Okay, so then what's done is, so I take my chromosomal DNA and get rid of the histones, add endonuclease called EcoR1. It's going to cut here, 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 and here. Notice by this fragmentation, I get a small fragment from beta globin, a medium-sized fragment from beta globin, and a large fragment from beta globin. That they were in mixture, but now they're separated by the gel electrophoresis and then transferred to this piece of paper. So on this piece of paper, there's this piece of paper once again. On this piece of paper, somewhere in the large region, there is a beta globin gene fragment that I can't see in my eye. There's a medium-sized fragment, and there's a small fragment that I just can't see, but I know they're there. Now the matter of detection. Okay, now pay attention here, because this is going to be hard for some students that have never been in the lab, and I have a hard time with this concept. So then the issue of detection. So what happens there is to detect the beta globin gene fragments. What I can do then is go to my laptop or whatever and do a Google search. Now, if you don't like Google, you do a Yahoo search. You don't like Yahoo, you do a Bing search. You just do a search, and you say, tell me the sequence of the beta globin gene. And within seconds, on this monitor in front of me, will come the sequence of the beta globin gene. And remember, it's a single strand, and it's a coding strand for beta globin. You must assume that unless they say otherwise, and you must assume it's the 5' end on your left. So there is a sequence of the beta globin gene. Okay, so once I see that beta globin gene, I get on the phone. And I call a company. I say, hey, company, yeah, you there? Okay, good. So what I'm going to do is I want you to make me a radioactive probe that's complementary to the beta globin gene. And the receptionist on the other end of the phone is going to say, do you have money? And I'm going to say, yeah, I got money. And she says, good, let's talk turkey. Read off the sequence. Because you know, money talks. Maybe you didn't know this, but money has a language of its own. So I get on the phone, I say, yeah, I got money, and so I'm going to read off the sequence. So I read off the sequence, and I say, no, it's 5' A, T, C, G, C, T, T. So I read it off to her. And sure enough, she writes it down, and then that day, there are machines that then make a radioactive, usually with P32, a radioactive probe that's complementary to one of the strands of the beta globin gene. And sure enough, by the next day, by FedEx or UPS, will come to my doorstep, or I could actually do this in the lab, but if I got a company can do this. So by next day, by FedEx, will come to my doorstep a radioactive probe, as long as my American Express is not over as long as my wife didn't use up all the money shopping yesterday. But in any case, oh yes. Anyway, so the idea is, then the question becomes, so next day will come the beta globin gene probe. Now here comes the hard part for some students. The probe, now suppose the probe that I purposely had made does not span the entire beta globin gene. The radioactive probe does not have to be equal in size to the gene of interest. It's complementary and anti-parallel to a portion of the gene of interest. Now, shown in the red line, suppose this represents the red line, the horizontal line, that represents the probe that some company made for me that's radioactive. And the probe now, because it's complementary to one of the two strands of the beta globin gene, what's going to happen is, notice how the probe will base pair with a portion of the small fragment, all of the medium-sized fragment, and a portion of the large fragment. And the probe will not base pair with any other fragment in that mixture, on this gel. It will not base pair with any other fragment. Why? Because it's not complementary to any other fragment other than the beta globin gene fragments here. Because it was made that way. So now here comes the hard part for some students. If a probe base pairs with part of a fragment. Notice here how the probe base pairs with part of the large fragment and part of the medium-sized fragment. If a probe base pairs with part of a fragment, it has detected the entire fragment. It knows where the fragment is on a gel. One more time. So the probe does not have to base pair with the entire fragment to detect it. If a probe detects part of a fragment, it has detected the entire fragment. Now that's what has some students have a hard time understanding, because again, it's more of a lab issue, but you must understand that. Now I was trying to think, what's the best way to explain this, and using an analogy. Now to use an analogy here, see this finger right, see this hand right here, see this thing right here, the ring. See this ring? This ring has been on that finger for 40 years. This ring has been on the ring for 40 years. Now, before my wife put this ring on my finger, suppose she put a GPS device in this ring. She put a GPS device in this ring. Okay, so then and then let's assume tomorrow or tonight, let's assume I want to go walking around or something like that, or I go to another city to do the Kaplan lectures or something like that. So like a couple weeks from now, I'm going to Grenada, SGU down there to do the lectures. But in any case, suppose as I walk around at night, my wife is sitting on the computer right here in front of me, and she's monitoring some satellite in outer space that's detecting where I'm at. Because in outer space, there's a satellite that sees that GPS device in the ring. So as I walk around, because that satellite instrument is seeing part of me, the ring, it has detected all of me. It knows where I'm at as I start walking around. So that's the same principle here. Same principle is that if a probe sees part of a fragment, it has detected the entire fragment. And again, some students have a hard time understanding that. But the idea is all you're doing is you're detecting a part of a fragment. If it binds base pairs a part of a fragment, it has detected the entire fragment. Okay, now what's coming up here is, let's come back to this figure. So here is the piece of paper that has those bands. And one of the bands will be a large fragment from beta globin, medium-sized fragment, large fragment coming from beta globin that I can't see with a naked eye. So then I add the radioactive probe, as shown in the red, I had made for me. I add the radioactive probe, and this is a process called hybridization. Hybridization, base pairing. So when I add the probe will now, so I take this piece of paper and put it into a flask or a dish in solution. And in solution, I then add the radioactive probe. The radioactive probe will now look at every band on that piece of paper, looking to hybridize. And the only place it can base pair is the large, medium, and small fragment that's shown in the solid lines. It base pairs with a large, medium, and small fragments coming from beta globin. Okay, so then after you do the hybridization, you take this piece of paper, you wash away the excess probe, and then you look at this piece of paper and you still see nothing. Why? Because radioactivity is colorless. So then what happens? Now, those of you who are radiologists will understand what I'm getting at here. Then what I do is I take this piece of paper that has the radioactive probe base paired with the beta globin fragments. I then overlay it with a piece of X-ray film. And then you let it sit overnight, and anywhere where the beta globin probe sees the beta globin fragments, it's going to strike the X-ray film. You come back the next day, develop the X-ray film, and on the X-ray film, what will be seen are three fragments in this example. A large fragment coming from beta globin, medium-sized fragment, and a small fragment coming from beta globin. Why? Because that's what the probe detects. The probe determines everything in this type of DNA blot. The probe determines everything. It determines what's being detected here. Okay. Is that clear? You must understand what I just discussed. As long as you're paying attention. Okay, you got it out? Okay, yeah, good. Thumbs up. All right, good. Yes. Okay. All right, now what's coming up is Gia, don't worry about how long it takes to the automatic. how long it takes to develop X-ray film. They're not going to ask you that on boards. Don't worry about that. That's detail you don't have to know. Don't worry about that. Just understand the concept here. How long it takes and how, what X-ray film you use. Who cares? It's just the concept here. All right, now what kind of blots are out there? And the kind of blots that are out there is, again, this is the process of hybridization. The kind of blots that are out there are listed here. These are the main ones. There are other ones as well. Now, I know I saw some of you type in already. First aid talks about snowdrop and all that. Forget about it. That does nothing for me. Snowdrop. Okay, snowdrop. Okay, it's snowing outside. I got to shovel the snow and it's dropping here. Who cares? That doesn't make you a better physician, understanding what snowdrop means. Understanding what the blots are and what they do. That's important to know. And these are the major blots here. And the first one that you must know, I cannot emphasize enough. You must know the name of the blot and what is being analyzed and what the kind of probe is being used. My wife will answer that. She's probably contacting some GPS seller. Sorry about that. I don't know. Okay, now she answered it. I told her, I said, answer the phone when it rings because the students will get upset with me and they won't come back. And I said, well, she said, well, hey, get a life. The phone rings, okay, big deal. Tell them, slow down a little bit. Okay, anyway, the bottom line is you must know the name of the blot and what's being analyzed and what the probe is being used. Now, what I mean by this is on board exams, if they say to you, a southern blot was used to do this or that, immediately you got to know DNA is being analyzed. If they say to you, a northern blot was used to do this, you think to yourself, immediately they're talking about RNA being analyzed. If you're sitting there thinking, oh, anything snowdrop, the N and snow, is that RNA, is that D? You're wasting valuable time. That does nothing for you. The idea is just you must master this. It's not hard. It's not hard. Just give you got a beautiful mind. Just use it. Just use it. I'm asking you to use your mind. So again, the first one here is the southern blot. And what is widely used in molecular medicine, widely used. And what a southern blot is used for is analyze DNA fragments after electrophoresis to separate the DNA fragments on the basis of size. And how are the fragments being analyzed? Using a radioactive probe, DNA probe. Matter of fact, on the previous figure, let me go back one figure. This on the right side is a southern blot. DNA fragments are being analyzed after electrophoresis using a DNA probe. This is a southern blot. Now, the next kind of blot to talk about is the northern blot. Now, a northern blot is where RNA is being analyzed. And how's it being analyzed? Remember, RNA is never fragmented. Never fragmented. But RNA is separated on a basis of size using electrophoresis, and the RNA is being analyzed using a DNA probe. In immunology, you talk about the Western blot, where Western blot is where proteins are being analyzed after electrophoresis using, and how they're being detected using, as Dr. Muscatella talked about, iodinated or enzyme-linked antibodies to detect those proteins. By the way, if you were want to know, I see on the public chat pot where the names come from. Well, what happened was the first person who discovered this kind of blot, his name was actually Southern. His name was actually Southern. So they decided to name the blot after him. If I discover a new technique or a new planet or whatever, I can call, my name is Pluto, I'm going to call the new planet Pluto. Because it's my right to call whatever it is if I'm the first discoverer of that. If I discover a new country, if I discover America and my name was Americus, what's the guy's name? I can't remember the Italian name. I'm Italian heritage. I forget his name. But in any case, they called it America because after some guy who named it after that. So you have the right to name whatever you discover. Okay. So his name was Southern. It's to analyze DNA fragments. Anyway, the next one was the northern blot, and so where RNA is being analyzed, and they try to be cute. So biochemists have this weird sense of humor, and they called it the northern blot. Not because some guy's named Northern. I mean, there are many people named Northern, but they were just named the same thing with Western, because the Western was just named after the person, not named after the person, but named after trying to be cute here. Okay, so all right, so then so the idea is, so the Western blot is where proteins are being analyzed after electrophoresis using iodinated or enzyme-linked antibody that Dr. Moscatello talked about. Now later on in genetics, today's Wednesday, so by Friday, at the end of the chapters in genetics, we're going to talk about the dot blot. The dot blot is a very powerful technique, genetic testing technique. It's where RNA, DNA, and proteins are being analyzed, and the big difference between a dot blot and the other three is no electrophoresis is needed. In other words, how powerful the dot blot is. I can take some of your DNA right now, and by tomorrow, I can determine whether you're homozygous, heterozygous, or normal for sickle cell anemia. This is one of the most powerful techniques out there, the dot blot. And because it's so powerful, the National Board loves it. Now, in your profession, the dot blot is only being used for a few diseases so far. Because you have to know, to use a dot blot, you have to know what the mutation is. We know what the mutation is in sickle cell. And in genetics coming up on Friday, we'll talk about the issue of hemochromatosis. We know what the mutation is, the precise mutation is in hemochromatosis. So we can use the dot blot to figure out genetic status. So the dot blot in your profession is only being used for maybe a half a dozen diseases, something like that. Not that many. But it is an up-and-coming technique in your profession. Okay, and again, there are other blots out there, like the Southwestern blot, and things like that. I doubt they're going to ask you about that. So, don't get too concerned about those other ones. Forget about the other ones if you don't know these ones, right? If you know these ones, you master these, okay. If you want to know what a Southern blot, okay, you can look in first aid. But master these ones, certainly. These are the ones you're likely to see on board. So you might see some of the other ones. There aren't that many, but these are the four you must master. Okay, so the first one we're going to talk about here is we're going to talk about the Southern blot. Now, first, by the way, where are we going with these blots? So in Chapter 7 in molecular biology, we're going to be talking about the Southern blot and the Northern blot. And in immunology, you already talked about the Western blot, so I won't do that. So that was discussed by Dr. Kim Moscatello. No need for me to do that. She talked about it in the Western blot. And in genetics, 2 days from now, we'll talk about the dot blot. So all four blots will be covered, and you must master them and understanding what they do, how they're used. Okay, so the first one we need to review is the Southern blot. Now, the Southern blot is very widely used. It can be used in genetic testing for many reasons. For example, in forensics, if you watch, like my wife watches that NCIS, Miami and Los Angeles, and I guess the other cities and so on. But when they say to you there's a crime scene and somebody's DNA is being analyzed, how was that DNA analyzed? And it's the Southern blot. Is how the DNA is being analyzed. So the Southern blot can be used for person identification in crime scenes. The Southern blot can be used for other reasons in genetic testing of diseases, as we'll also talk about in this chapter. So the first thing I want to talk about here, though, is the use of the Southern blot for person identification. Okay. All right. So this is now, again, the Southern blot, one more time, is where DNA fragments are being analyzed after electrophoresis to separate them in the base of size using a DNA probe. Okay, so here is now the use of the Southern blot. Now, the Southern blot is used as the basis of a major technique called RFLP. You're likely to hear the words RFLP or letters RFLP on national boards, but the Southern blot is its basis. Now, what does this mean? Now, again, look away from this. Don't look at this figure. Just think about this a minute. Just close your eyes, just think. What this is about is, remember, you have 23 chromosomes that you got from your mother, and you have 23 chromosomes that came from your father. And but they're not identical chromosomes in sequence. So some of the chromosomes you got from your mother are different, slightly different in DNA sequence compared to the chromosomes that you got from your father. You may not know this, but that you and I are 99.9% identical in DNA sequences. I mean, it's amazing. The 23 chromosomes that you got from your mother is 99.9% identical to the 23 chromosomes that I got from my mother. It's amazing, huh? That 0.1% difference is why you are you and I. Because that's a point, that little bit of difference is what causes all this differences between us as human beings. Here, okay. Now why is this? How can that be taken advantage of? And the idea here is, if I did a biopsy of you and put that biopsy into one test tube, and did a biopsy of your neighbor and put it into a second test tube, and a biopsy of me and put it into a third test tube, and then get rid of all the histones with detergents, and in each of the three test tubes I added the same endonuclease, what we would find is that DNA fragments that come, the lengths of the DNA fragments that come from you are different than the lengths of the DNA fragments that come from your neighbor, and different than the lengths of my DNA fragments, and different from everybody else in this world of 7 billion people. Unless, of course, you have an identical twin, then they'd be identical. But other than that, the lengths are different from everybody else in this world. Look at the letters. L stands for lengths, the restriction fragments. Restriction fragment lengths. The lengths of your fragments are different from everybody else in this world. And it's due to that 0.1% difference in your chromosomal DNA sequence compared to me, your neighbor, and everybody else in this world. Here. Okay, and because of that, we can use it for person identification, the lengths of these fragments, because the lengths of your fragments are going to be different from everybody else in this world. Okay, so now the idea here is, again, I'm trying to take this a little slow, and those of you that had this in the lab, I apologize for boring you, but I want to make sure students understand this. Okay, now the idea is, why? Why are the lengths of why do you have the lengths of the fragments different from everybody else? And the reason is because of this 0.1% difference. But what causes the difference? What's this 0.1% difference due to? And the answer is, there's two reasons that give rise to the lengths of your DNA fragments, the sequences. And one reason is due to mutations. The other reason we'll talk about at the end of the chapter. So one of the reasons why the lengths of your DNA fragments are different from me is because of mutations that I might have that you don't have, or mutations you have that I don't have in our chromosomal DNA. Now, what I mean by this is, suppose what you're looking at right here is a portion of a very long chromosome that goes far to your left and far to your right. You're just looking at a portion of a very long chromosome. And suppose this version of the chromosome, we'll call this version A, chromosome A. This chromosome A is the chromosome that you inherited from your mother. Remember, you inherited 23 chromosomes from your mother. Whatever particular chromosome number it is, chromosome 1, chromosome 3, chromosome 5, it doesn't matter what number it is, whatever that particular chromosome is, the version of it, we'll call it chromosome version A. So don't get bogged down. It's a particular DNA sequence of one of your chromosomes that you inherited from your mother. Okay, now in chromosome A that you inherited from your mother, in that one vicinity, suppose at 3 sites, at site 1, 2, and 3, there are 3 palindromes for ECO-R1. At site 1, there's the GAATTC. At site 2, same sequence. At site 3, another sequence. The rest of the nucleotides not even shown here. Again, envision this chromosome is 4 nucleotides in some very long sequence. And somewhere in it is GAATTC, found at site 1, at site 2, and at site 3. Okay, and that's if you inherited chromosome A and you got it from your mother. So therefore, if one or both of your chromosomes, whatever chromosome number it is If one that you got from your mother and the one you got from your father are both chromosomes A, then what's going to happen is when you add equal R1, it's going to cut here, here, and here, as shown by the purple lines, vertical lines. And suppose on board exams they tell you the length of this fragment from site 1 to site 2 is 1.2 kb, kilobases, 1,200 kilobases from site 1 to site 2. Where'd that number come from? They tell you that on board exams. They say to you the length of that fragment that's cut between site 1 and 2, they tell you on boards it's 1.2 kb, 1,200 nucleotides from site 1 to site 2. And suppose they tell you the length of this next fragment from site 2 to site 3, suppose they say to you it's 0.9 kb, 0.9 kb, 900 nucleotides from site 2 to site 3. How do you know that? They tell you that on board exams. Okay, now that's the chromosome that you inherited from your mother, and suppose it's the normal chromosome. But suppose the chromosome that you got from your father, the same chromosome number that you inherited from your father, suppose it's a slightly different version. In other words, a slightly different DNA sequence. And because it's slightly different, we'll call that chromosome version B. It's the same chromosome number, but a slightly different version. Now, why is that? Why is it a different version? Now, the reason for this is, suppose, as an example, see this A here at this palindrome at site 2. See this A? Suppose that A is converted to a G. In other words, DNA polymerase did not pause and check and correct itself. In the ancestry of this chromosome that you inherited from your father, whether it was 500 years ago or whether it was 1,000 years ago or whatever, somewhere in the ancestry of your father, this particular chromosome got a mutation in it, where the A was converted to a G. This is what's known as single nucleotide polymorphisms, sometimes called a SNP. Okay. Now you're going to say, wow, man, doesn't that mean I'm going to have a disease and pathology? Isn't Dr. Barone going to be talking about this issue? And the answer is, no, you don't have to have a disease here. And the reason is, remember, now we almost coming stuff we talked about earlier. Remember, 98.5% of your chromosomal DNA is junk DNA. You can have all the mutations you want, like you see right here, as long as it's not in a gene. If it's in a gene, it causes sickle cell. If it's in a gene, it causes PKU. If it's in a gene, it causes galactosemia. If it's in a gene, it can cause G6PD deficiency. If the SNP is within a gene. But you have so much junk DNA that if it occurs in the junk DNA, it causes no pathology. Absolutely no pathology. This is why. Here comes the why question. In case somebody asks you, why do you have so much junk DNA? And the answer is sort of like a protection. Now, you don't have to know this number, but the mutation frequency of DNA is something like 20 nucleotides per generation. In other words, the 23 chromosomes that you inherited from your mother, you have 20 mutations that your mother didn't have. And your offspring, if they inherit those chromosomes, will end up with 20 mutations that you didn't have. One more time. So from generation to generation, in your 23 chromosomes that you got from your mother, you have 20 mutations in them that your mother didn't have. And then you're going to pass on 23 chromosomes to your progeny. And that progeny is going to have 20 mutations that you didn't have. So in successive generations, there are mutations. 20 mutations coming from your father's side and 20 mutations coming from your mother's side. So and that's what gives rise to the 0.1% difference of our chromosomal DNA. 0.1% of 3 billion nucleotides is quite a number of nucleotides. But the idea is, so as long as the mutations are occurring in junk DNA, causes no problem. That's why you have so much junk DNA. It's like a protection. When these mutations come, hopefully it's going on in the spacer DNA, causing no pathology. Okay, everybody understand that? Okay, everybody understand that? Okay, that's why you have so much junk DNA. You think, oh, that's just wasteful. It's not wasteful. It's that when these mutations come, you're trying to protect your 20,000 genes. Now, if it happens in a gene, it could give rise to pathology. Okay, so again, the chromosome B that you got from your father is a slightly different version. So don't think genes here. But now, because that A was converted to a G in your father's chromosome that you inherited, that's no longer a palindrome for ECO-R1. No longer a palindrome for ECO-R1. So if you have chromosome B, that ECO-R1 will cut here. So it's going to cut here and here, but not here. So let me ask you this. So then what's the size of this fragment? What is the length of this fragment if you have chromosome B? What's the size of this fragment if you have chromosome B? That's right. It's 1.2 kb, 2,100 nucleotides from site 1 to site 2. If one or both your chromosomes is version B. All right. So then, now it says over here, is only one of the blot is correctly corresponds to the genotype displayed. In other words, suppose your genotype of these two particular chromosomes is genotype AA, AB, or BB. In other words, you have two identical chromosome A's, or you may have two identical chromosome B's, or you may be one chromosome A and one chromosome B. All right. So only one blot corresponds to this particular blot. Now, the idea is detection of the fragments, the 1.2, 0.9, and the 2.1 kb fragment. Here comes the probe. Remember I told you the probe determines everything in a southern. This is a southern blot. The probe determines everything. So as shown by, now suppose the probe spans from here to here. The probe was made on purpose to span, to hybridize with the chromosome from here to here. Okay, so as shown by the vertical lines, that represents hybridization. This probe, now notice, it base pairs with part of the 1.2 kb fragment, not all of it, but part of it. But because it detects part of it, it detects the entire 1.2 kb fragment. It knows where the 1.2 kb fragment is on a gel or a piece of paper. The probe detects the 1.2 kb fragment. So let me highlight it there. So if one or both your chromosomes is chromosome A, the probe will detect the 1.2 kb fragment. Yes or no? Will the probe detect, this particular probe, will it detect the 0.9 kb fragment? Yes or no? Will the probe detect the 0.9 kb fragment? And the answer is no. Why not? Notice here, the site 2 is right here. The probe is not base pairing with any part of the 0.9 kb fragment. The probe is not base pairing, not hybridizing with any portion of the 0.9 kb fragment. So the 0.9 kb fragment is not detected by this probe. Not base pairing with any portion of it. Okay, you got that. So again, you got to understand, I'm taking it extra slow here. Now in chromosome B, suppose one of your chromosomes is chromosome B. So therefore, you add the probe, and again, the probe was designed to base pair with this portion of the chromosome. So as shown by the vertical lines, the probe will base pair with this portion of the fragment. And remember, if the probe base pairs, hybridizes with part of a fragment, it has detected the entire fragment. So that probe now will base pair and recognize the 2.1 kb fragment and no other fragment coming from chromosome B. Okay, is that clear? Okay, has everybody got that? You must understand this. The probe determines everything. And if a probe base pairs with part of a fragment, it has detected the entire fragment. It knows where it's at on a gel or a piece of paper. All right, now suppose they give you the southern blot. They say to you, only one of these are 2 or southern blots. DNA fragments are being analyzed after electrophoresis using a DNA probe. Now suppose the genotype is AA, AB, or BB, AA, AB, or BB. Which of the 2 southern blots is the correct one using this probe? Let me see your answers here. Think about it. Excuse me. That's right. It's the one on the right. This is the one on the right is the correct southern blot. Now, what I mean, now watch the arrows. Watch the arrows. Now, suppose my genotype is AA. I have two identical chromosome A's. One came from my mother and one came from my father. There was no mutation there. Two chromosome A's. And so therefore, that when I do a cut the DNA with equal R1 and then subject the DNA fragments to electrophoresis, and then on this lane right here with the AA genotype, there are many bands from all the chromosomal DNAs. There are hundreds, if not thousands of bands in this one lane that I can't see with my eye. I don't see anything. But then when I add the probe, the radioactive probe, that base pairs with this portion of the chromosome, notice how the probe will detect the 1.2 kb fragment and no other fragment. So I have the AA genotype if both my chromosomes are version A. Okay, now you can watch the arrow. Okay, so there it is. So here's the 1.2 kb fragment, and it's shown here in this southern blot. Here. All right, now suppose my genotype is AB. Okay, let me erase this arrow. If my genotype is AB from chromosome A, here is the 1.2 kb fragment, right? From chromosome A, it's got to be a 1.2 kb fragment. But by the way, notice that the intensity of this band is less than the intensity of this band. This is a thick band. This is a thin band. If I have an AB genotype, why? Let me see your answers here. Why is the AA, you have a very thick band, and over here, a very thin band? Why? Why? No. That's right, because why a thick band with AA genotype? Because I have two chromosome A's. I have twice the copies of the 1.2 kb fragment. So the intensity of the band is a relative idea of how much is there. So there's twice the amount of chromosome A than there is in the AB genotype. So this is a thick band and this is a thin band because I only have one copy here of the 1.2 kb fragment and two copies here in the AA genotype. Now from chromosome B, here is the 2.1 kb fragment. There is the 2.1 kb fragment. Again, it's a thin band. So if both bands are seen, that means my genotype is AB using that probe. And then lastly, if my genotype is BB from chromosome B, here is the very thick band seen at 2.1 kb, a thick band because there's two chromosome Bs, two copies, therefore twice the amount of band here than there is over here detected by the probe. So that is if the genotype is BB. So again, I want to stress here. Let me stress as much as I can. In a southern blot, the probe determines everything. You must understand the concept of the probe. What is the probe doing? It's detecting a portion of a fragment that comes from a chromosome. And if it base pairs with part of a fragment, it has detected the entire fragment. Now, what I'm going to do here is, it's not in your book, but what I'm going to do is I'm going to erase the color in the middle. What I want to do is demonstrate how important this thing is. Probe determines which DNA fragments are being seen. I'm going to illustrate this or prove this. What I'm going to do is in the middle of this figure, I'm going to erase the color, okay, and use another probe. Okay, I'm going to watch now. I'm going to erase the color. Okay, there is nothing new except the middle part. So if I have chromosome A, the endonuclease equal R1 will cut here, here, and here, generating the 1.2 kb fragment and the 0.9 kb fragment. Nothing new. If one or both chromosomes is chromosome version B, it's going to cut at site 1 and at site 3, but not at site 2 because of the SNP, the mutation. If I have chromosome B, if one or both chromosomes is version B. All right, now let's use a different probe. Okay, here comes. Now, this is where you get a chance to work this one out on yourself. What would this southern blot look like if I use this probe? In other words, it was designed on purpose to span from here to here. Okay, I'm going to give you 15 seconds to think about this, and then I'll give you the answer. Think about it. All right. And the answer is it's the southern blot on the left is the correct answer. Now, how do we know this? Okay, now get ready with yes or no. If one or both chromosomes is version A, will that probe detect the 1.2 kb fragment? Yes or no? Let me see your answer. And the answer is yes. It detects the 1.2 kb fragment. Because if the base pairs are part of it, it detects all of it. Will the probe detect the 0.9 kb fragment on a gel or piece of paper if you have one or both chromosomes, chromosome A? And the answer is yes, because if the base pairs are part of it, it detects all of it. It detects the 0.9 kb fragment. And if one or both chromosomes is version B, will it detect the 2.1 kb fragment? Yes or no? That's right. All you got to do is look for the Y. That's right. Yes. So it detects the 2.1 kb fragment. All right. So now Y is the southern blot on the left, the correct answer. Now watch the arrows. Watch the arrows. If the genotype is AA, so the person has 2 chromosome A's, then the probe will detect the 1.2 kb fragment. It's going to be a very thick band because you have 2 copies of it, and also detects the 0.9 kb fragment. If it detects part of it, it detects all of it. So very thick bands because you have 2 copies of the 1.2 and the 0.9 kb fragment. Now, if the genotype is AB, from chromosome B, here is the 2.1 kb fragment. From chromosome A, here's the 1.2 kb fragment. From chromosome A, here is the 0.9 kb fragment. 3 bands are being seen because that's what the probe detects here. But notice the bands are thinner because you only have 1 copy of that of the respective chromosomes. And then lastly, if the genotype is BB, from chromosome B, there's going to be a very thick band at 2.1 kb. Very thick band. Twice the amount here, then over here if the AB genotype, because you have 2 copies here and 1 copy there. Notice this is the southern blot. It's not hard. You can't memorize this. You have to think about it. It's not hard. It's not hard if you think here. Now, if you have time tonight or tomorrow, just do this one more time. Just practice this once or twice or whatever. And then, in other words, just erase all this stuff here. Again, the probe determines everything. I cannot emphasize this enough. And to prove this again on your own, do this on your own. Use another probe. In other words, suppose the probe spans from here to here, shown by the green arrow, from here to here. And you're going to get another southern blot that's going to be correct if the probe spans from here to here. Just do it again. Use another probe, and then you'll get the, okay, now I understand how you interpret a southern blot here. Okay, now what we're going to do is one more figure, then we're done for the day. Okay, so the southern blot can be used for person identification, like you see here on genotypes. But the southern blot and the RFLP, that technique called the RFLP, the basis is the southern blot. The RFLP can be used for genetic testing of disease. And the disease we're going to be talking about here, major disease for detection, is sickle cell anemia. Now, on board exams, sickle cell is probably one of the highest yield diseases that you're likely to see on board exams. You got to know everything you can about sickle cell. One of the top 10 of all diseases for boards. So they might ask you the pathology or how you treat it with hydroxyurea and all these other things, pharmacology. But they might ask you a question about sickle cell regarding genetics, regarding detection, and that kind of stuff. So that's what this is designed to talk about. Now what happens here is it says in this vignette. So this is the last figure we're going to do today. So in this vignette, I'm not going to read the sentences, the first few sentences, but what it says is in the vignette. It says a man and a woman have a son who has sickle cell anemia. They have a daughter who has not been detected for carrier status, but phenotypically normal, and the woman is pregnant. So on the boards, the likely question is they want to know about the status of the fetus. It's a typical board question. They'll show you a pedigree, and they might want to know what is the status of the fetus. All right, let me ask you this. We'll talk about genetics tomorrow, but what's the probability? Get ready now with your response. What's the probability that the fetus is a carrier for sickle cell anemia. Based on the pedigree alone, what's the probability the fetus is a carrier for sickle cell? Not 25. No, not 25. It's 50% probability of being a carrier. 25% probability of being homozygous with the disease, and 25% probability of being homozygous normal, but a 50% probability of being a carrier. Why? Because you must know, without them telling you, that sickle cell is autosomal recessive. So what that means is the father and the mother both are carriers for the trait. Both of them have a defective beta globin gene. The other gene they have is normal, but both the father and the mother are heterozygous carriers of the trait. How do you know that? Let me ask you this. How do you know that? How do you know the father and the mother are carriers for the trait? Don't worry about, no, we're not talking about bands here. I'm talking about the pedigree alone. Don't worry about the bands. How do you know? That's right, because the son has the disease. The son has the disease. The son is homozygous for sickle cell anemia. In other words, the son must have inherited one of the defective genes from the father and one of the defective genes from the mother. Remember, the son is inheriting 2 chromosomes that have the beta globin gene. And if both genes are defective on whatever chromosome number it happens to be, don't worry about that, but they inherit both chromosomes that have defective beta globin gene, the son has the disease. So the pedigree alone tells you the father and the mother are carriers for the trait. Now remember, on board exams, they don't have to show you in a pedigree who, in an autosomal recessive disease, who is a carrier. Carrier meaning half open, half closed circle, half open, half closed square. They don't have to show you heterozygous carrier. They don't have to do that. You must know the father and the mother are carriers because the son has the disease. That's how you know. And therefore, because the father and the mother are carriers, that means in a Punnett square, 50% probability of being a carrier AS for the trait. Okay, now that was the old days. And that's pedigrees and probabilities and Punnett squares and all that kind of stuff you talked about in genetics. That was the old days, where all you can do is make predictions. Probability, 50% probability of being AS, 25% probability of being SS, and that kind of stuff. But now, the power of molecular biology, the power of the RFLP using the southern blot can determine exactly what the outcome of the fetus is. The beauty of the RFLP. All right, now that's what the bottom part of the figure is. All right, and that says, let's continue reading. It says, the mutation causing sickle cell anemia. That position, remember we talked about this earlier. There's a glutamate at position 6 is converted to a valine. Remember, and that was due because an A was replaced by a T, a missense mutation at position 6, resulting in an A replaced by a T. Therefore, the codon for glutamate converted to the codon for valine. All right, and now look what the rest of the sentence says. It just so happens that mutation destroys, look at the word, destroys, eliminates a palindrome restriction site, same thing, for an endonuclease called MST2. Now, these endonucleases are often shown in italic with a Roman numeral. Don't worry so much about that. But what the palindrome is not important. The words are important. It says that the mutation destroys a palindrome for MST2. All right, now what does that mean? What that means is, in this diagram, suppose in the orange, this represents the beta globin gene in some chromosome. Now, the beta globin gene, you don't have to know the details, but it has 3 exons and 2 introns. That's what the orange color represents and the yellow color. So 3 exons and 2 introns. Now, suppose on board exams, they tell you that in the vicinity of the beta globin gene, there are palindromes for MST2. Okay, and as shown by the arrows, suppose they tell you there's a palindrome over here, outside the beta globin gene, there's a palindrome right here, and there's a palindrome over there. So as a result of fragmenting of the beta globin chromosome, we get a 1.15 kb fragment and a 0.2 kb fragment, a very small fragment called 0.2 kb, 200 nucleotides in length. Now that's the information they would give you, they might give you on board exam. They might give you on board exams. Now the vignette says that the mutation causing sickle cell destroys the palindrome, destroys this particular palindrome. And as a result, if one or both of the chromosomes has the sickle allele, the MST2 will cut here and cut there and not there. So what's the size of the fragment if one or both of the chromosomes is sickled? And the answer is it's 1.35 kb, because this DNA was not fragmented. Not hard, as long as you're thinking, as long as you're following me, as long as you're thinking, not being distracted, you're following here. So the palindrome was destroyed by MST2. Okay, the palindrome was destroyed, so MST2 cannot cut it because it's no longer a palindrome. Okay, so now, so what's happening here is DNA is now taken from all family members. Just do a biopsy of all family members, including amniocentesis of the fetus. So you take DNA from all family members and put them into 5 separate test tubes. And in 5 separate test tubes, we add detergent to get rid of histones, and then each of the test tubes we add MST2 to cut up the DNA. The DNA is then subjected to electrophoresis to separate the fragments on the basis of size. There are hundreds or thousands of fragments in each of these lanes that you cannot see with a naked eye. So now how did you detect the beta globin issues, the beta globin fragments? The vignette tells you. Here it is. It says that you always ask what probe is being used. Remember I tell you the probe determines everything. It says in the vignette, it tells you. It says a probe, right here, a probe detects the 5' UTR. Remember yesterday I told you better know what UTR is. Isn't it a perfect example? The probe detects the 5' UTR of the beta globin gene. And that's shown here in the red. See this horizontal red line? That represents a probe that was made commercially, that on purpose, that detects not the beta globin gene itself or anything in the beta globin gene. It detects the 5' UTR of their beta globin. So now let me ask you this. So by using that probe, if one or both of the chromosomes is the normal chromosome, chromosome A, what size fragment will this probe detect? Let me see your answer. What size fragment will that probe detect? What size fragment? That's right. Well done, everybody. It detects the 1.15 kb fragment. Yes or no? Will it detect the 0.2 kb fragment? Will it detect the 0.2 kb fragment? And the answer is no. Why not? Because the probe is not base pairing with anything with the 0.2 kb fragment. So it detects the 1.15 kb fragment, but not the 0.2 kb fragment. Why? Because that's what the probe was designed to do. Not hard. Think. It was designed to see only the 1.15 kb fragment if one or both chromosomes is version A. Now, if one of the chromosomes is or both of them are version B, what size fragment, S I should say, what size fragment, if one of both chromosomes has a sickle allele, what size fragment will it see? That's right, everybody. Well done. It detects the 1.35 kb fragment. Because if the probe detects part of it, like you see right here, it detects all of it. So the probe detects the 1.35 kb fragment. So now we go to the southern blot. And again, this is a southern blot because DNA fragments from all family members are fragmented with MST2, subjected to electrophoresis to separate the fragments on the basis of size, and then probed using a probe that detects the 5' UTR of the beta globin gene because it says so. All right, now what this means is any fragment that's 1.15 kb in length, or size I should say, is indicative of a normal allele. Any fragment that's seen that's 1.35 kb in size is indicative of a sickle allele. Okay. All right, now let me stress here. Let me stress before we move on, because we're almost done. It's another minute or two. I'm going to stress that on board exams, they are unlikely to give you this information right here. More than, and all this information down here that you have to deduce. More than likely, they will not do that. What we're doing here is explaining the sizes of the bands, where they came from. on board exams, what they're likely to do is give you a pedigree and give you a southern blot, and you analyze the southern blot to determine the fetus. You don't need this information. I'll show you. You don't need this information. You see the information because I'm explaining what's happening here with the bands. Now, what I mean by this is, suppose, and the way you interpret the southern blot, suppose this information was not shown, normal allele or sickle allele. Suppose it was not shown, just blot it out. Now, so you know the father. If you went to the father first, you always go to the person you know most about genetically on a southern blot. One more time. To interpret a southern blot, you always go to the person you know most about genetically in a southern blot. Now, what I mean by this is, if you went to the father first, the father has two bands. And you know the father is heterozygous because the pedigree says so. But you would not know if you went to the father first which of these two bands is normal and which one is sickle. It says so over here, but if you didn't know that information, you would not know which of these two bands is normal and which one is sickle. You always go to the person you know most about genetically. Let me see your answer. Who is the person you know most about genetically? That's right. It is the son, the boy. That's right. That's right. What can you tell me about these two bands right here? This band right here. And the answer is, that's right. It's got to be what? It is SS. That's right. This son is homozygous. They have a thick band, two overlapping bands that are the sickle fragment. Yeah. So two overlapping bands. It says so right here on the right side, but you don't need this information. You just always go to the person you know most about genetically. Now, once you do that, you make the assignments of the bands. Again, the thick band, because it's two identical bands, both of them all have the mutation. Then once you determine that, then you back off and say, where were they inherited? How were they inherited? One of the bands came from the mother, and one of them came from the father. And sure enough, now we can go to the father, and the upper band in the father got to be S. And the other upper band in the mother got to be capital XS as well for the son to have the disease. And therefore, the lower band in the father got to be A, because the father is heterozygous. How you know that? The pedigree says so. And the mother's lower band got to be capital A, because the pedigree says so. Not that this says so over here on the right, it's just the pedigree says so. Okay, so now that we make those assignments, we can now go to the fetus. Here it is. So now look at the fetus. What can you tell me about the upper band? Is it S or is it A? That's very simple. And the answer is it is S. And we can tell you about the lower band. It's got to be A. And so now what is the status? Here it is. What is the best conclusion about the fetus? Let me see your answer. What's the best? There it is. That's right. It's got to be a carrier. The fetus is a carrier. 100% certainty. 100% certainty. None of this probability and none of that kind of stuff. The southern blot, as well, the basis of the RFLP, determines exactly the status of the fetus. Now, it says in the vignette, the daughter does not have sickle cell, but may be a carrier or might be homozygous normal. But notice the southern blot tells you exactly. The southern blot tells you the daughter must be capital, must be AA. Because one coming from the mother and one comes from the father. So again, look how beautiful the southern blot is, the RFLP. They're beautiful how this is. Now again, now let me stress here. We're done for the day, and tomorrow in the first hour, we're going to finish off this chapter. At the end of the chapter, you're going to see another example of a southern blot that you do on your own. And you'll see it's not that difficult as long as you think. You can't memorize this stuff. I'm telling you right now, you can't memorize this stuff. You have to think your way through this. It's not hard as long as you try it out. So we're done for the day, and then tomorrow we're going to talk about the northern blot. That's measuring RNA. Then we're going to talk about the PCR reaction for paternity testing. Some baby's born, you want to know who the father is, and that kind of stuff. That's coming up tomorrow in the first hour. And then we'll do a couple of vignettes, and then we're done with molecular biology. After that, we're going to get into medical genetics and chapter 1 of medical genetics with all those pedigrees and so on, and these different definitions of pleiotrophy, imprinting, locus heterogeneity, all coming up in chapter 1, a high yield chapter. So we'll get into that, and then maybe the Hardy-Weinberg issue tomorrow. So a lot of genetics. But if you fear genetics, give me a chance. Give me a chance to explain it. That's all I ask. Just give me a chance. Okay, so we're done for the day, and I'm gonna post on the Facebook. I'm gonna post 3 more questions based on tonight's today's or yesterday's lectures. And again, please do it. Explain your answer. More people, the better. And if you see somebody writing in the wrong answer, help them out. Explain, no, that's wrong. You don't have to be sassy or anything. Just be nice. Just help each other out. And then late tomorrow afternoon, I'll give you the answer and so on. So again, it's to your benefit to do these type of things. Okay, everybody, had a great time tonight. So I look forward to seeing you tomorrow promptly at 6 o'clock Eastern time. See you guys. Take care. Have a great evening.